Got it कर देना इसको.
Got it. Got it. Got it. Recording शुरू हो गया.
Okay. So now we are about to start.
So good morning, everyone. Welcome you all in Day 2 International Conference. So again, welcome you. I request to our volunteers to start the session. Sanjana and Ananya, please start the session.
Recording हो रही है ना बेटा?
Hello.
Thank you, ma'am. A very good morning to one and all present here. I welcome all the delegates, our esteemed guest speakers, Professor Dr. Srinivas Subramaniam, the Scripps Research Institute, Florida, Dr. Jitendra Narayan, Sir, Senior Scientist, CSIR IGIB, New Delhi, Dr. Ilyas Rashid, Sir, Scientist, Tata Institute of Genetics and Society, DBT in STEM, Bangalore, respected committee members and the participants to the second day of the International Conference on High Throughput Data Analysis and Artificial Intelligence in Biological Research, organized by Amity Institute of Biotechnology, Amity University, Uttar Pradesh, Lucknow campus. Thank you. Yesterday, we delved into the cutting-edge advancement in data analysis, artificial intelligence, and their application in the field of biological research. Today, we have another exciting lineup of invited lectures and oral presentations that will continue to expand our knowledge and inspire us to push the boundaries of research. We would now extend our heartfelt invitation to Director, Amity Institute of Biotechnology, Professor Dr. Janmejay Kumar Srivastava, sir, to welcome our guest and address the audience.
Okay, thank you, Ananya. I extend a very warm welcome to all the guest members, speakers, faculty members, and attendees. Today's session has to be started by Dr. Srinivas Subramaniam, and I think he had joined already. So, Dr. Subramaniam, welcome to this session. And let me introduce Dr. Srinivas. He is currently working as a professor in the Department of Neurosciences, UF Scripps Biomedical Research, Jupiter, Florida, USA. And he has obtained his PhD in 2004 from University of Heidelberg, Germany, where he worked on the molecular signaling that triggers neuronal death at Johns Hopkins University and he has also worked for his postdoctoral work at Johns Hopkins University, where he addressed the mechanism of tissue-specific dysfunction, focusing on the Huntington's and Parkinson's disease. He has also extended his experience in his current work at the Scripps Institute, and he has over 27 years of experience of teaching and research in this area, and he has already guided around 15 students. So with this, I again invite Dr. Srinivas Subramaniam for his specialized lecture. Welcome, sir.
Well,
welcome,
thank you.
Srinivas.
Thank you very much. Thank you very much. Can you all hear me?
Yes.
That is great. So I should go and start my screen, right? I guess. Well, I want to thank you, Prekshi, first, and then all the organizers of the meeting, because this is an unexpected opportunity for me to be here. And I really should say that I never gave a talk in the middle of the night at 1 o'clock. It's the first time I'm doing that. So,
Yes.
I should really, it's an opportunity, I think. It's great to know people and their research and also share my work. And again, I want to thank Prekshi for this opportunity. So let me now, can you see my slides, please?
Yeah. It's
Okay.
visible now.
Okay, so my audio is clear. You all can hear me.
Yeah, that's also.
Okay. Okay, so, what I'm going to talk today is some of the new work we are doing on Huntington disease, and I will show some of the high throughput data that we have currently submitted for publications, and this data highlights the importance of how mitochondrial translation is impaired in Huntington disease. So you're welcome to stop me if you want to ask any questions or any concerns. You can message me so that I can read and answer your questions if needed. I'm trying to move the slide. Yeah, so pretty much we agree that the way we function in the world it depends on how our brain is interacting with the world. We get the data from the surrounding and then we process them in the brain and then we respond. So pretty much everything begins in the neurons and ends in the neuron. This is what this cartoon predicts. So unfortunately, this beautiful organ has experiences many kind of diseases, which has been described here. Unfortunately, we do not have a therapy for any of these disease. So a lot of research needs to be done in brain areas to understand the mechanisms and ultimately design and develop new therapies. So one of the area that we are focusing on is to apply a multiple interdisciplinary approach to understand the disease mechanisms. So we use cell molecular biology and in vivo system approaches to gain insights into the brain diseases and then develop potential therapies. So today I'm going to talk about Huntington disease, which is a genetic disorder which affects you can see here is the patient who has a diagnosed for Huntington disease. And these patients have a problem in motor coordination, ability to control the movement, and they are not able to even hold things. This is one of the major symptoms of Huntington disease, where there's uncontrolled movement, unable to do fine motor skills, which we all take it for granted. So disease affects people of all over the world. So although it's a rare disorder, this can impact the quality of life dramatically and affect, and most people die prematurely within 10 years of a diagnosis of the disease. The good news is that we know the gene. This is a autosomal dominant disorder, 100% penetrant, and the gene involved is a single gene. It's a monogenic disease. Gene is called Huntington. So Huntington is a long protein, consists of more than 3,000 amino acids. And the problem lies at the N-terminal part of the gene. The N-terminal part of the gene codes for CAG repeats. Normally, we have around 35 CAG repeats, which give rise to glutamine, polyglutamine, but whereas Huntington disease patients, they'll have more than 36. And that results in adult onset of Huntington disease. And the disease also affects children, and the children will have further expanded version of CAG repeats. They can have up to 121, and this again, a larger the expansion, earlier the disease symptom and the death. So the disease, this has been like more than 40 years. We know that this is the gene which causes the death. And if you look at the pathology, this is the MRI image of Huntington disease patients. Here you can say on my right, and this is the lateral ventricle of the brain, and that is the striatum. So striatum normally is intact. You can see that's gray area here. Next to this black region, that's striatum, and that is the cortex. So in Huntington disease patient, the striatum degenerated predominantly. Then there is a cortical atrophy. Interestingly, the cerebellum seems to be not impacted by Huntington disease. Here is the problem. The disease-causing gene, Huntington, is present everywhere in the brain and also peripheral tissue. And it predominantly damages the striatum followed by cortex. So we still do not completely understand why does this selectivity comes from and what are the mechanisms involved in this selective degeneration. So here is a further emphasis on how this disease is so selective to the brain. Here is the patient who has undergone longitudinal magnetic MRI, and they did it for 4 years. And within the 4 years, you can see the striatum shrink substantially. This is 2008, and within 4 years, there is a substantial loss of striatum. There is little effect on the cortex. Almost nothing happens to the cerebellum. So when striatum degenerate, what happens? The striatum is a kind of center of your brain functions, I would say. Center in the sense, it's a circle where you get information from other regions like cortex and also substantia nigra. So this region has a striatal cells, which consists of 2 different receptors, and they work collecting the information from the major areas of the brain, which is cortex and substantia nigra. Then they send signals down the pathway and then through other brain regions, and then this signals goes to the cortex. Then the functions are carried out. What kind of function the striatum carries on? Motor function. You saw the patient that they have difficulty in controlling their action. So if the striatum is not functioning, and then there is a problem in executing motor function and decision making. You may decide that today you want to have some nice Indian lunch. So that decision making should also happen in the striatum. And the motivation to come to this seminar, this presentation also happens in the striatum, and also reward and reinforcement. So striatum is critical for our day-to-day life. And when that is not functioning and degenerates, it impacts our actions. But the question again that why does Huntington promote striatal specific damage? Or how does it even promote cellular dysfunction in the striatum? So obviously the gene has been described like some long time ago. Many mechanisms have been proposed. like aggregation, autophagy, mitochondrial dysfunction, transcription dysregulation, apoptosis, excitotoxicity, so on and so forth. But this again does not explain why does the striatum degenerate. So for today's talk, what I'm going to show you is that we focused on a mechanism where the potential, the translation may have impacted in the striatum. So how did we come to this? Is a long story. We have been working on striatal signaling pathways, which we published in 2014. But as you know, the idea came from the study. You all know the translation where the mRNA releases from the nucleus, and the mRNA is now undergoes translation with the help of tRNA, and each tRNA starts decoding the codon and mRNA and start making protein synthesis. Well, this is the fundamental properties of the cell. Every cell carries out translation and then decodes the mRNA and start making proteins or peptides. So interestingly, in Huntington disease, not much studies have been done on the impact of translation per se. So we were kind of interested there. And then we recently made some progress on this area. So for today's talk, I have two things to tell you. What I'm going to show you that the ribosome seems to have stalled in Huntington disease. So they go much slower. Much slower in making the protein. And another interesting finding is that particularly the mitochondrial translation seems to have impaired in Huntington disease. So which I'm going to discuss a little bit detail. How did we arrive at this conclusion? So the bottom line is that the ribosomes move at a certain speed, speed with the involvement of many other factors like tRNA and the secondary structure of the RNA and so many different factors. So they move one codon at a time and slowly starts making the protein. What we found is that this speed is inhibited or regulated by normal Huntington, not the expanded version, normal Huntington, like up to 35 CAG repeats. But this speed could be further inhibited by mutant form of Huntington. If there is an expansion of CAG, or if there is a polyglutamine, like in Huntington disease, that can block the movement of ribosome and ultimately inhibit protein synthesis. How did we arrive to this? If you're not aware of the fundamentals, I can go very quickly. You know, ribosomes, we have two different kinds of subunits, major subunits, 40S subunits and 60S subunits. They're basically a bunch of proteins and RNAs. They come in contact with the mRNA and they scan the mRNA as a single monosomes, and then they start moving on to the mRNA. Each mRNA can have multiple monosomes, which are called polysomes. So as they travel, they start making the peptides. So interestingly, this different portions of these ribosomal subunits can be visualized biochemically. So for that, we take the cells and we run it through ultracentrifugation in a sucrose gradient. The sucrose gradient can separate different subunits. Like the lighter one, 40S will come on the top, followed by 60. And the monosome, which is basically consists of 40 and 60, then the di, trisomes, and polysomes. So you can really separate this onto the gradient fractionator here, which is mentioned here. And because this fractionator can use UV to detect the RNA, you can see this kind of peaks which appears as a final output. So this final output can be highly qualitative as well as quantitative. So in other words, if you have a larger density of polysome, which is mentioned here, so you will have a hope you all can see this portion. So there is this huge density of polysomes can be seen on the rightmost part of this graph, and that shows that there's a very active translation going on. So one can measure the translation status by looking at this graph and comparing it to the control group. So the first thing we did is to do that, we need to have a well-defined cell lines or cells, and then we can go from there. When you do protein synthesis work, it is better if you have a homogeneous cell type so that we can surely attribute the changes that we are seeing to that cell. So luckily we have these cells called, these are striatal cells. This is the control cell. They have 7 CAG repeats in Huntington. And we have Huntington disease cells, which they have either 2 copy of mutant Huntington up to 111 CAG repeats, or they may have only 1 copy, it's heterozygous, 1 copy of mutant Huntington, which is 111 CAG repeats. So we basically have 3 types of cells in this case: wild type cell and the 2 types of Huntington disease cells, one with 2 copy of Huntington and 1 copy of Huntington, which are referred as Huntington HD het and HD homo. So we just run the gradient on this cell, which here I'm just showing you only homo for simplicity, because we know homo is exclusively of Huntington. What you can see here is that this is the monosome peak, subunits under the monosome and then the polysome. What you can readily see is that in mutant Huntington, the polysome density is much, much higher compared to the control. So when you quantify this, you can clearly see that the ratio of polysome to monosome, which is an indicator of how strong is the ribosome binding to the mRNA, elongating mRNA during translation. So that seems to be very high, highly significant increase. Then we thought, wow, then this means probably there is a lot of translation going on in Huntington disease because there's a huge polysome. But it is not so. Here is the data. When we run protein synthesis experiments on these cells, which is mentioned here, triplicates of control and triplicates of homo, you don't see much protein synthesis, which is measured using, in this case, radio-labeled methionine, which is methionine is amino acid incorporated into the proteins. Most proteins has methionine. So we can visualize the methionine incorporation as shown here. You can see that the protein synthesis is decreased in this cell, even though they have a bigger polysome. That means there are a lot of ribosomes bind to the elongating RNA, but they're not translating. So we have published this work, and we did many more experiments to confirm that the ribosomes or too much ribosomes on mRNA in Huntington disease but they are not translating. They have the potentially the traffic jam of ribosome in Huntington disease. This is a very new study, and we were very excited. And here is an further more data on this finding. Here we use puromycin. Puromycin is the tRNA analog, and it's getting incorporated into the ongoing translation. And then you can identify puromycin using antibody. Here is the immunocytochemistry of puromycin incorporation in control cells. You see control cells have a robust incorporation shown by high intensity. And the het and homo, they have a substantially decreased intensity of puromycin incorporation, confirming that the protein synthesis is much, much lower, even though they have ribosomes being actively recruited onto the mRNA. So, as you all know, that the translation in cells happens at two different sites. One in the cytoplasm translation, another one is mitochondrial translation, which I'm going to come back again. But what we wanted to see is that, is there any effect of translation in the mitochondria as well? So here is the data. So what this data shows that there is a substantial decrease in this protein synthesis in het and homo cells, as you can see here. Here is the quantification, but also there is a substantial decrease in the translation in the mitochondria as well. And this is a quantification of that. So despite so much work has been done, how the cytoplasmic and mitochondrial translations are coordinated remains unclear. So our data shows that in Huntington disease, both cytoplasmic translation is decreased, as well as the mitochondrial translation is decreased in Huntington disease. Then what we did is we wanted to go further onto this in mitochondrial translation. We isolated mitochondria from Huntington disease cells, and then we incorporated with puromycin with it. See, in vitro conditions, we can see any changes in translation. Yes, even in vitro condition, incorporating protein synthesis and Huntington disease mitochondria is substantially diminished. So many of you are aware of that. In mitochondria, there are 13 transcripts. Mitochondria provides us only up to 13 transcript made by mitochondria, and they're getting translated. These 13 transcripts are part of electron transport chain complexes, and they're very critical. With the 13 OX40 transcript translated protein, there are a bunch of proteins coming from the cytoplasmic translation. So these two, one from the cytoplasmic mitochondrial nuclear encoded mitochondrial protein, and then the mitochondrial encoded mitochondrial protein, they come together and they form the complexes to carry on fundamental function. So because there is a substantial loss of protein synthesis in Huntington disease, we wanted to know more about the ribosomes. Okay, how does the ribosomes are arranged or moving on these transcripts? So for the first time, we applied a technique called ribosome profiling in Huntington disease. This technique has been used in different models, but not in Huntington disease. So we were the first one to do this, and this is how we did it. So we cultured control cells, HD, het, and homo cells, and then we did many different steps. Basically, all these steps we separate the monosomes, which are indicator of that they're binding to the elongating mRNA, and then that we can identify, we can separate them running through the gradient, and through this gradient we isolate the elongating monosomes using various steps, including proper RNase treatment, etc. And then we now isolate the ribosomes and the RNA, and then we can do a ribosome, we can extract the ribosome protected RNA, and then we go for deep sequencing, and then we can identify what are the transcripts and where the ribosomes are on those mRNA. You know, it's a very tricky technique. It took us some time, but we were very successful. So for this particular talk, I'm going to focus on mitochondrial ribosome. So what we have found is that the result would look like typically like this. You will have I'm trying to show on the right side. Okay. So what you're looking at is graph is, these are the data will look like once you do all these different steps. These are totally bioinformatics. I think it's well suited for this particular symposia. And again, I'm not expert on bioinformatics, but this is so important for us to for this, and we collaborated. We made international collaboration to come here. So the data would look. This is for example, this is an mRNA of some gene. So the orange one is mRNA. You can see it also shows the UTR. Mostly UTR do not have ribosomes, particularly the 3' UTR, most genes. But then you can see the ribosomes sitting on this mRNA and these different exons. So this is how the data would look like, which I'm going to talk more about it. What did we find in mitochondrial ribosome sequencing? So surprisingly, what we found is all the mitochondrial encoded OXPHOS transcripts, they have a high ribosome occupancy, which again indicates that most likely these ribosomes are stalled. They're not able to move much, move like in control. Here is a little bit expanded version. So what you're looking at here is a gene called mitochondrial NADH ubiquinone oxidoreductase subunit 1, short form MTND1. This is a mitochondrially encoded transcript. The blue one is mRNA. So it has got only one exon. And then you can see this, the orange one is where the ribosomes are in control. So in Huntington disease cell, you see the ribosome, the mRNA levels are slightly less, but the ribosomes are very high distribution. So it seems to be accumulation on the 3' end of the transcript. With one copy of Huntington disease. With a 2 copy of Huntington disease, we have further reduction of RNA and also relatively less ribosome occupancy. So you know that when there is a RNA divided by the riboseq, the ribosome occupancy gives us the actual level of ribosome occupancy, which is kind of mentioned here. First, you take the ribosome protected fragment as a readout. You can see very high in it, and HOMO is less. I'll come back to that if you have any question, why less in HOMO. And then this is the RNA of the corresponding genes. And then you take a ratio of RFP and RNA, that gives the actual translation efficiency. So one should expect that the het should have a high translation turnover for this particular protein. and also the HOMO. Basically, in this case, what you're looking at is that the transcript, the MTND1 transcript, has a huge amount of ribosome sitting on them. And then if you do that, if you look for another transcript, which is MTND4, also you see a similar trend. There's a high ribosome occupancy in Huntington disease cells. And if you take another one, which is MDND6, also you see similar, and so also MDCO1. All these are mitochondrial transcripts. All 13 mitochondrial encoded transcripts have a very high ribosome occupancy. But if you look at the nuclear encoded mitochondrial transcript, mitochondrial protein, they do not have much difference. That's what is shown here. Here is an example for NDU. FE1 is a part of the subunit encoded by the nucleus, or cytochrome C1 protein, or cytochrome C reductase binding protein. These are all made by the nuclear mRNA nucleus, and they go into the mitochondria. So the ribosome occupancy on these transcripts is not impacted in Huntington disease. There is a specific effect on mitochondrial encoded OXPHOS transcript. Sorry about this picture. This is low quality. Then what we wanted to look is that high ribosome occupancy is really translated into protein. What is happening at the protein level? So for that, we took a mass spectrometry approach. We collected these same cells, isolated the mitochondria, and then we did a quantitative TMT proteomics. So we looked at the overall changes in the protein level on the mitochondria in control cells compared to Huntington disease cells. Here is the data. So what I'm showing you here is I'm comparing the ribosome occupancy of a protein, of an mRNA, and I'm showing the corresponding protein level. So what you're looking at here is on the left side, this is again MDNT1, has a very high ribosome occupancy in control, in HD. And then if you look at the protein level, there is not much difference in head, but protein level seems to be decreased in HOMO. Similarly, MTND2 had a similar pattern. There's a very high ribosome occupancy, but the protein level seems to not alter. Then if you look at SDHA, commonly used mitochondrial marker, the ribosome occupancy is less. And then the protein level seems to be not so much impacted. The steady state level of this protein is not impacted. Similarly as DHB, another subunit. Oops, this is bad. I'm sorry. This is this showing you the network of how these interactors, the protein that we identified as the network, they are very closely linked, and they work on mitochondrial pathway, translation pathway, and mRNA cleavage, etc., etc. I'm sorry. This file is corrupted. So here is some interesting proteins that have been found. For example, Cavin1 seems to be have no change in ribosome occupancy, but they have a very high protein expression in Huntington disease, particularly in HOMO. And there seems to be Huntington copy-dependent manner. For example, PTCD3, as the ribosome occupancy is decreased in head, further decreased, and the protein level also is decreased. And then there is PRXL2, another protein that has a very high ribosome occupancy in head, but they both HD have a substantial reduction in the protein levels. And then here you see opposite trend. They have a similar ribosome occupancy, but very high protein expression, steady state protein expression. So all this suggests that the mitochondrial translation is impacted at two levels, both at the ribosome occupancy level and also the protein level. So how this is controlled is exclusively by the ribosome movement, or other post-translational modification, post-translation changes are required, remains to be discovered. So here is the summary and the model. So basically, what I'm showing you here is that in the mitochondrial transcripts in Huntington disease have a very high ribosome occupancy, and how that is been, what are the mechanisms that happening there, whether the Huntington itself have a direct role in regulating the ribosome occupancy on these transcripts is still unclear. And what we are looking at is that there is a effect of ribosome movement in the cytoplasm by Huntington, whether those kind of changes impacts the translation on a mitochondrial transcript is not known. There could be some kind of signals which may be regulating, which are unclear. So this interesting dichotomy that of mitochondrial of OXPHOS transcript is the first time is reported, and we're very excited to see how this research goes further. So this paper is currently under review, and it is also part of this work is also on bioRxiv. You can look up if you're interested on this. I'll be happy to answer any question. Here is the people and the funding agencies who have been actively involved in these different projects. And I thank you for your patience and the opportunity. So I'll be happy to take any questions.
Hello.
Yeah.
I'm Dr. Rachna Chaturvedi. So just, acha, okay.
Okay, we
Ananya,
would now take a moment to introduce our expert session chair, Dr. Rachna Chaturvedi ma'am from Amity Institute of Biotechnology and Dr. Meenakshi Srivastava ma'am from Amity Institute of Information Technology, Amity University, Uttar Pradesh, Lucknow campus to chair this session. Dr. Rachna Chaturvedi ma'am and Dr. Meenakshi Srivastava ma'am are experts from their field. I would now request Dr. Rachna Chaturvedi ma'am to take over the session.
Thank you, Ananya, and thank you, Sanjana, for just giving me this opportunity to chair this session. So here we are done with the talk of Dr. Srinivas Subramaniam, and his topic of his talk is ribosome profiling and mass spectrometry reveal widespread mitochondrial translation defects in a statal cell model of Huntington disease. So thank you so much, sir, for your valuable talk. Thank you so much.
Thank you.
Okay, so sir, you have given just introduction to us one of the most trending approaches of high throughout data analysis, that is ribosomal sequencing. The evidence of widespread dichotomous effect on the ribosome occupancy and protein turnover of mitochondria related genes in Huntington's disease is surely a revolutionary approach in medical research. Thank you so much, sir, for this valuable talk. So now this session is open for question answer. If anybody need to just have any query for this topic, so you can just ask questions to sir. All the participants, this session is open for question answer.
Thank you, Dr. Rachna Chaturvedi ma'am and Meenakshi ma'am for your kind presence and chairing the session.
So anybody?
So now
Ayushi has a question.
So some hands have been raised. Okay.
Some hands raised. Somebody has raised hands for question. So you can ask your question here. Anybody?
Yes, Ayushi, you can proceed.
You need to unmute to talk.
Actually, Dr. Chaturvedi madam, because of the problem,
technical issue.
No, not technical issue. Actually, the participants, the noise was coming. So we have muted the participants. And the
So now
session-
you can unmute Ayushi, if it is possible.
I am requesting from the host, and till then the questions are in the chat box that can be read out by.
Okay, okay. We can see the question
Yeah, that can be read
here,
out.
okay.
Oh, okay. I think I can unmute them, right?
Okay. Sir, you can unmute?
Yeah, I think I did it. Yes.
Ayushi?
Prekshit.
No,
Oh.
madam. It is from the host. Sir, questions are in the chat box. Rachna ma'am, can you please proceed with the question?
So now I can read the question of Ayushi. Sir, how can mitochondria or any other cell organelle be isolated? What is its experimental protocol? So
Yeah,
sir, can give answer to
Ayushi, people have been
Yeah.
isolating mitochondria from 100 years. You can look at the protocol on the various legitimate sites. You can look at original papers. You can see a lot of papers, a lot of resources for that.
Okay, Ayushi? You got it?
Madam, I have requested for the unmute just in 2 minutes.
Okay.
Dr. Srinivas,
So
can you
anybody,
hold
if anybody wants, then you can just give a question in chat box,
Yeah.
so I can read out. If anybody has any questions, so Prekshi, sir, I'm reading question of Prekshi. Sir, I wanted to ask, what are the challenges of developing drugs that target ribosome stalling in Huntington disease?
Yeah, that's a very good question. I think currently we're doing experiments to look up any specific transcripts. Interesting thing is, Huntington itself have a ribosome stalling, Huntington mRNA. So we're looking at that aspects, like how, if you understand more about how the ribosomes are stalled on Huntington RNA, then we can design drugs to block Huntington production. That is our future idea.
Okay, Prekshi, got it? So again, I'm requesting, if you have any question, then you can write in chat box.
Excuse me, sir.
Yeah.
Now the participants are unmuted. They can.
Okay, Ayushi, now you can ask a question directly.
Okay, thank you, ma'am, and thank you, sir, for such an informative lecture. My question actually is that actually I was willing to know that nowadays through plastic surgery, it is possible to replace many body parts. So if any part of the brain is getting damaged due to some genetic disease or anything, then is it possible to artificially develop that part I mean the artificial neural system can be developed, an artificial part can be developed and can be transplanted into the brain. I mean, I know maybe it's not possible right now, but can it be possible in future?
Yeah, future, anything can be possible, right? Humans always have hopes. That's how we evolved. We always look for better future and better conditions. So the surgery, people are trying to develop models where they can implant iPSC-derived cells, which can make new neurons in the brain. Those research is ongoing, but we don't have any strong data that can be used for HD patients right now. Yes, in future it's possible people may replace the dying neurons with freshly made neurons. That can be possible, but I don't think, if you're thinking that to replace the whole brain as a surgery, like a kidney replacement, I'm not sure we are very close to it.
Okay, sir. And sir, just one more question I have. Sir, gene therapy is possible, right? So why treatment using the gene therapy are rare cases? I wanted to know the answer to this question.
Yeah, I'm not an expert on gene therapy, but there are a lot of questions, a lot of clinical trials have been stopped because the targeting is a problem, right? Gene therapy sounds great, but where do you target? Because this disease affects the deep brain. Gene therapy has been successful on peripheral cancer biology, where the disease affects the blood, but gene therapy is not very well executed for a brain disease because of the targeting issue. Specific targeting is a problem.
Okay, so here one question again from Ananya Singh. So Prachi, can you unmute Ananya?
Yes, ma'am. Yes, ma'am. Sure. Ananya, I think you are unmuted and you can open your
So
video
you can ask a
also.
question directly.
Okay.
Sir, so my question was, what are some potential implications and applications of AI in diagnosis and treatment of Huntington's or any other neurological diseases?
Well, a lot of things going on that. I think people are trying to see with Huntington disease, it's already obvious because it's genetic disorder, right? You can do a genetic screening and have HD. The AI has been used to identify potential biomarkers. Again, the area is not well developed, and there's a lot of interest on that because so far I think they have used biomarkers which not done by AA but by vigorous research that they can use some cytoplasmic structural protein, I forgot the name, as a marker. But again, a lot of things is unknown. AI has been used to study the progression of the disease, probably to look at the MRI images. But again, a lot of things are not well, not to the point that we know a lot through AI research.
Okay, Ananya, I think you got it. So anybody else for questions?
Thank you so much, sir.
Okay, anybody else for questions?
Yes, ma'am. Can I ask with the due permission of chair?
Yes, definitely.
Okay. Thank you so much, ma'am. First of all, very heartfelt thanks, Dr. Srinivasan, for accepting our invitation and such a short notice and giving such a nice talk, which is really very new for me also, because we are working on the transcriptome analysis, we are working on the microRNA sequencing analysis, but the first time I am hearing about the ribosomal sequencing. Okay, so, what are the difference? Is there any procedural or protocol difference between the transcriptome data sequencing and ribosomal data sequencing?
Oh, yes. There's a lot of differences that you need to use. The kind of RNAs that we use to chop off the ribosome binding, the chop off the ribosome bound RNA. The RNAs are different. And also the isolation of the fragments on the gel. We need to isolate the RNase treated fragment on the gel, which is critical point to look at exactly the ribosome location. So it's not an easy thing like a transcriptome, which is anybody can do, even high schoolers can do. But riboseq sequencing requires some standardization, and it's a very sensitive technique. And more than that, it's very expensive. So
That's what I was asking, because very recently we are getting certain papers, and when I saw your talk, I was quite promising to learn about it. Sir, one question is that when you are performing the RNA sequencing, you have to filter out the proteins because most of the time protein synthesis is going on with the ribosomal section. So, how do you manage all these things? And is there any if we use Illumina or Roche 450 or Nanopore for the transcriptome sequencing, is any such kind of platform available for the ribosomal sequencing so that not only me but my audience can be benefited by this answer?
Yeah. I think we have published a established protocol, and a full protocol is published. You can look at that. You can separate the protein because we're isolating the 13 nucleotide fragment running on the gel. So when you run a gel, all the proteins are off. So you look at the 13 nucleotide region and then gel extract that region, a region which is where the ribosomes are bound. So I'll be very happy to share any other information apart from what we published. So yeah, it's a very interesting to see of course, RNA expressions are not translated into protein. You need to also look at ribosomes, how the ribosomes are sitting. So there's a lot of research shows that the RNA expression not directly related to the protein expression, whereas the riboseq can reflect the protein expression. But again, it varies. In our case, there's a more ribosome, but there is no protein production. So we think the ribosomes are stalled under traffic jam.
Okay. Thank you so much, sir.
Okay.
And I will be happy if you share your paper, if you don't feel any reservation in sharing your paper, which you have published as a protocol. And
Yeah, it's a published, right? You as an open access. So.
It is open access. Okay.
Yeah.
Thank you. Thank you so much.
So
Thank you,
anybody
Dr.
else?
Rachna ma'am, for giving
Okay,
me opportunity.
thank you so much, Prachi. So anybody else for any question? No. So just I am partially concluding this session because we are already having 2 speakers more in this session. So thank you so much, sir, for your nice and valuable talk. And I think all our participants must be benefited from this important talk. So thank you so much, sir.
Thank you, Prekshi. Thank you, Prachi, and then Dr. Rachna. Appreciate your time.
Okay, thank you so much. Thank you so much.
Bye-bye. I'll see you later. Bye.
Thank you and so much for in odd hours you accepted our invitation and given the nice talk.
But it's nice though. I can go for a.
So sir, so we can do good night to you, sir.
I can go for a walk, early morning walk now. Thank you.
Srini sir.
Nice to
Prachi ma'am.
Yes, ma'am.
Srini sir, which particular tool you all suggest to the researcher to use at the beginning of their research? Like which AI-based tool?
Well, that's what I'm asking Prekshi to help me. She's not, she hasn't come back to me. It's been a month.
Prekshi, why that so?
I'm not permitting. Okay, I'm so sorry, sir. She was highly inducted in the conduction of this conference and for just execution of this event. Later on, very soon she will be in touch with you, sir.
No. I was joking.
From my part.
Then I'm also waiting for your answer, okay?
Apologies from my side, from chairs and from speaker.
Thank you so much, sir.
Okay, thank you so much. Thank you so much to all.
Thank you, guys. Bye.
Thank you, sir. Good night.
Have a rest of the nice meeting. Bye.
Okay.
Thank you,
Thank you
Rachna ma'am.
so much,
Thank
sir,
you
for
so
such
much.
an insightful and informative session. Moving forward, we would now invite our next guest speaker, Dr. Jitendra Narayan, sir. Sir is currently serving as the senior scientist in Big Data and Informatics.
Ananya, sorry, I'm interrupting you. Let me confirm Dr. Jitendra Narayan has joined or not. Let me confirm. Just give me 2 minutes.
Ayushi, please lower your sound.
Jitendra, can you open your camera, please? Dr. Jitendra Narayan.
Just wait a second, okay? Give me a second. Yeah, hi.
Sorry for the inconvenience. I would now like to invite our next guest speaker, Dr. Jitendra Narayan, sir. Sir is currently serving as a senior scientist in Big Data and Informatics Unit at CSIR Institute of Genomics and Integrative Biology. Sir has received his PhD from Austria-West University, Wales, UK, and his postdoctoral from Laboratory of Evolutionary Genetics and Ecology, Belgium. Sir's research mainly focuses on development of computational pipeline and algorithms to harness the huge amount of biological data in genomes, transcriptomes, and proteomes to answer specific biological questions pertaining to this domain. Sir has published several research articles in various prestigious peer-reviewed journals and has over 2,000 plus citations. Sir is also granted several patents for his pipelines. We are truly blessed to have Sir with us today as our guest speaker. I would now request our session chair, Dr. Meenakshi Srivastava ma'am, to take over the session.
Thank you, Ananya. Jiten sir, I welcome you on behalf of the organizers. Please start your session.
Okay, so I do not, okay. Yes. So hi everyone. My slides is visible to everyone or not? Hello? Hello?
Not right now. But
Yes,
yes, now it is visible. It is visible now, sir.
Okay, so am I audible to everyone?
Yeah, you are audible and you are at 12th number of slide.
Okay. So, so hi everyone. I am a computational biologist, and I mostly work on comparative genomics and genome analysis work. Most of my work is on NGS analysis. I also work in AI and ML section as well a bit. So I will discuss today neurofibromatosis, the one that I am actually working right now. And we are working to solve the specific problems that I will discuss in next slide. So this is actually Okay. So this is actually genetic disorder that affect the nervous system. So this neurofibromatosis is actually a genetic disorder. So that some mutation happening in chromosome number 17, and that leads to a big problems in the lifestyle of the infected individual. Whereas it also linked with the development of a benign tumor called neurofibromas. So it links, it is directly linked with the neurofibromas and brain, spinal cord, and nervous system and skin related diseases. So the things that it happen is autosomal dominant disease. So it's actually a multi-system disorder and is prone to develop benign and malignant tumor of CNS and peripheral nervous systems. So most of the times, whenever someone is having neurofibromas, they tend to develop some benign and malignant tumors in their neuron system. These are caused by mutation in the NF1 gene. So especially, this is also called an NF1 mutation gene. So the mutation happened in NF1 gene leads to neurofibroma development. And the most common of them are neurofibromatosis. So usually this belongs to a rare genetic diseases where most of the time, 1 in every 3,000 individuals are reported to have this kind of mutation. Most of the problem that I see is many Indians, especially in Indian population, there are many people suffering from NF1, but they are not having any updates. I mean, they are not aware of it that they are having NF1 mutation because they're not going under any mutation genetic testing, or they do have some symptoms, but they ignore it completely, and it leads to a big problem in the future. So that's the reason that I started working on this direction and trying to see what is all about. So the first signature of neurofibromas are actually café de lutte, where we have small patches on our skin. So this is the first signal, first thing that we noticed in neurofibromatosis diseases. So the spots you see here. So if you look at this spot in my cursor is visible. Okay, no, anyway. So the spots on the skin. So this is very common in India, and you see many people are having spots in their on the skin spot. There are many patches on the skins, but they are not aware of it, that they are suffering from certain mutation, and they can be it can lead to many diseases, many weird situations in future. So but the problem is there are many other diseases as well that is associated with these patches. So we were not able to differentiate between them. Is this NF1 or is something else? We don't know. It might be possible someone is having patches and it's not NF1, is something else. So there should be a specific software or tracking system that can easily track the mutation and tell you that this belongs to NF1 and not spread one, for example. So there is no such software that can differentiate between these diseases. So we decided we started working in this direction to develop a model specific algorithm. Sorry.
Please mute.
Yeah, hi. So there is no specific software or something that can differentiate between these NF1 mutation and spread1 mutation, for example. So we decided to work in this direction so that the user will be aware that these kind of patches actually belongs to NF1, not spread1 and other diseases. So apart from that patches, there is one more basic signature is the list nodule in our eye. So if someone scan your eye, it can easily see the nodules in your eye, and they can inform you that you're suffering from certain diseases or not. But these are not the only signatures that classify that you belongs to NF1. It might be possible you have a long hand or you are tall. It might be possible it links with the NF1 as well. So we started digging into it, that do we have any other symptoms that can tell us that someone is suffering from specific diseases? So when I started exploring the domain, we noticed that not only the spots that we are actually interested in the very first round, that someone is having the patches on their skins, it might be possible they are having that specific mutation, but apart from that one, learning disability as well. So if someone has a learning disabilities, it also linked with the NF1 as well. So if someone has an NF1 mutation, they will certainly going to have some learning disabilities.
Dr. Jiten,
in future.
Dr.
Yeah,
Jiten, can I interrupt you?
Yeah, sure.
So, sir, Jiten, please on your camera.
Sure.
Okay.
Oh, sorry. Okay.
Okay. Thank you so much.
So if someone has a learning, if someone has a learning disabilities, it's also linked with the neurofibroma. But learning disability is not only linked with the NF1. It could be having something else as well. So we certainly need to combine all the learning disabilities, listener deals, fracklings, neurofibromas, skeletal abnormalities. So we need to combine all together into one complete module and design a specific algorithm so that we can identify and differentiate the NF1 to the rest of the diseases. So mostly the learning disability is very common in other diseases as well. So we certainly cannot use only learning disabilities as a symptom for NF1 classification. The simplest example is skeletal abnormalities is also very common in other diseases as well. So we certainly cannot, but we certainly need to combine all together into one so that we can in future, we can simply tell them that this guy is suffering from NF1 and they need to be taken care by the medical experts. So moving to the next one. So, Okay, so genetic mosaic. So in addition to that one, there is one additional is genetic mosaic, especially where we have an mutation in a specific cell type. And so it looks to the mosaic pattern in your entire body, and it also linked with the NF1, and somehow we need to classify them that this is a specific mosaic signature or not. So in this case, is we certainly need a specialized AI method that can easily classify them. So I actually with the help of our students, we dig into the specific signatures that we have, and we notice that list nodules is only present in 95% people, or more than 95% people, whereas caffeine is present in most of the people. 99% of the people have these patches. But in addition to that one, there is other interesting fractings, other patterns that we noticed is present. So early childhood cases, we have list nodules. So it might be possible someone is suffering from specific list nodules problems, we can easily detect them in early childhood. Whereas if someone has a caffeine spot, we can certainly identify them. But we need to sub-classify, and that is what we are doing right now. So here is deletion problems that are also noticed that so NF1 microdeletion symptom is characterized by more severe clinical presentation that majority of NF%. So deletion is the one cases where once someone has a deletion in NF1 gene, it leads to a big problem, like facial dysmorph, severe global development delay, cognitive disabilities. So there are many things happens, tall stature, many things happens once someone is having deletion, microdeletion in NF1 gene. So we started noticing that someone is having tall stature and long hand, they are somehow linked with NF1. So we are using this information in our algorithm so that we can simply classify in one go. So the deletion that I discuss here in deletion one is large stature and feet is very common once someone is having deletion, whereas large facial dysmorphic features are also common once having some deletion in NF1 gene region. So these kind of things are easy to track with the help of computer vision, and we can simply classify them that this are problem of microdeletion or just a point mutation. So we can easily observe and classify them. So to this one, I summarize all into one this picture with the help of my students. So these are eyesight scanning. So we will simply recommend everyone do the scanning of your eye, then scanning of your patches, and combine all together to sub-classify them. Okay, so the proposed model that we are working right now, so this is just a proposed one, and we are trying to upgrade and modify over the time, and we are improving our model right now. So the resource duration, so we are in the phase one when we use all these, we scan all these PubMed journals and try to find out the specific features and the features that we can use for the model training. We done this one. We're developing an algorithm, so we are still under progress. We're developing algorithm to identify the specific to use the specific features, curated a list of the features that we collected from the PubMed papers, and then we use them in the algorithm and implement them and integrate in the clinical data. So in addition to that one, we also collaborated with EMS to receive some data, and we are going to add the clinical information as well to combine them, and then we go for the testing and effectiveness testing. So we are still in the phase 3 and 4, and we are improving the model, and we'll do the validation in the last stage. So that's all that we are doing right now, and we'll be happy to add more features in it in future. Thank you. Thanks, everyone. Thank you. Hello. Thank you.
Yes, thank you so much, Jiten sir. I hope our students must have learned a lot. And which, again, I would like to know, any students, do you have any query? Students? Wait a minute, sir. Just a moment, sir. Sir, on which particular tool you are working?
Actually, I am using the camera vision softwares, and the tool will actually take the images of the patients and then sub-classify the type of the patches we have in our body.
Okay. 1 minute, sir.
Yeah.
Okay. And sir, while you classify, is there any specific algorithm which has given you the best result?
Oh, okay. So we are still in that, not in that phase. We are improving our data first, because we also need to add the clinical information as well. So we are collaborating with AMS to get some clinical information. So once we have the clinical information, we'll test it. So we are still not in the testing phase
That,
yet.
that process.
Yeah.
So you
Yeah.
are still not able to comment that this algorithm has given you the best result. Okay.
Not in a situation. Yeah. Thank you.
Okay. Thank you, sir. Yes, Dr. Prachi.
It was very nice talk, Jitendra, and
Indeed,
thank
very
you so
nice.
much.
Very nice.
And I would like to say from the platform, the man of algorithm and languages, Dr.
True.
Jitendra.
Yes, I asked him that how is, like, he's still waiting for the clinical results so that he can comment that which algorithm has given the best result.
Yeah.
We
Yeah.
are waiting.
Yeah.
We are also waiting with you, sir.
Yeah. Thank you.
And, I think the both platform from the IT and biological background people are interacting together. So Dr. Jitendra is really working such a nice for the algorithm development for. So Jitendra, I have one question.
Mhm.
Will you people also go for certain validation aspect, and how would you proceed for the validation aspect of your algorithms? And
Actually, so once we have the trained model, we'll use the real clinical information that someone is suffering from NF1 mutation. And for example, I mentioned in my talk that someone has a tall stature and long hand. It's one signature that might be possible that NF1 is an NF1 mutation happens in that individual. So we'll simply incorporate the clinical data and we'll run our software. The software say this is predicted, is yes, is more likely suffering from some certain disease, and we will test it using the clinical data. That what does clinical information say? Does it say that is positive or negative? So we'll use the clinical information to validate our algorithm.
So one of our students, Ayushi,
Mhm.
wants to ask something. Yes, Ayushi, please unmute yourself. Thank you, ma'am. Sir, actually, you mentioned that this is a genetic disorder, right?
Mm-hmm.
So, sir, as far as I know, one of the symptoms that I can see on the screen is the show up of the learning disabilities.
Mm-hmm.
So, as far as I know, if children show learning disabilities, then mostly, I mean, through special education and through various techniques, they can overcome their learning disabilities in normal cases. But if these learning disabilities are due to genetic causes, then I think they will pursue these learning disabilities for lifetime, or the learning disabilities can be cured
No,
through special
I will say that learning disability is just delayed. Is not. I mean, learning disability. Someone is learning. Some kids are able to learn in age 4, but some will start in age 5. So it's 1 year gap. Learning disabilities. Okay. So someone started learning a specific ABC at the age of 3. Somebody started at the age of 4. So it's just a delay in the learning. So especially in our cases, we notice that if someone has an NF1 mutation, they will have a delayed in learning. So they will start ABC learning started in the age of 4 or 5. This is what we noticed in our literature search, that mostly they started learning ABC at the age of 3 or 4, whereas normal kids start at the age of 2 or 3. Because the learning is delayed. That's one line. Yeah.
Okay.
Thank you for the question.
Thank you, sir.
I have one more question, Jitendra.
Mhm.
As I have just, because of the technical glitch, just skipped 1 or 2 slides, but that I can discuss later on.
Mhm.
Like you have taken the neurofibrostoma disorder.
Mhm.
So can these kind of model, what you are generated,
Mhm.
be used for other disorders which are showing the near about similar symptoms, or it is exclusively
Mhm.
for,
So the
because in
overall,
many of the, because in
yeah.
many of the neuronal disorders these
Mhm.
kind of symptoms are appeared.
Mhm.
So
So the overall idea was to classify them. Like this is the one problem that we see, that we were unable to differentiate between the first and second disease. So we need to have a specific algorithm that can easily classify them. For example, learning disabilities, as the question is raised, like if someone is having learning disability, doesn't mean that he is suffering from NF1. It might be possible he's having ADHD. So ADHD also have learning disability symptoms, right? Whereas we in NF1 person, if having NF1 mutation, they also have a learning disability as well. So this is what we wanted to classify. So if someone has a patches and learning disabilities, then he's more likely to be NF1 positive than ADHD, because ADHD do not have patches. So we need to certainly classify them. So this is the one that I'm mentioning, that if something is very similar, we cannot classify them because the mutation, because the pattern is same. So we certainly need a help of computer to classify them. And if once I have a model ready, I can simply we can tweak for the other diseases as well. So if you want to know that can we apply the same algorithm to other diseases as well? Yes, we can apply by after tweaking the algorithm. Yeah, thank you.
great. So I think you are continuing your presentation, or end up one micro.
Okay.
Ah,
So do we
Excuse
have
me,
any
sir.
question? Mhm.
I'm sorry to interrupt, but I have one more question, sir.
Yeah, go ahead.
Sir, you mentioned that in this disease,
Mhm.
no spots are visible on the skin.
Mhm.
So, sir, I mean, very rarely, I mean, someone who is the expert of neuroscience or neurology can only think of, if there is the presence of spots on the skin, then it may be this disease. Otherwise, most people would tend to think that if there are spots on the skin, then it may be a skin disease. So, is it a fact that many a times this disease is mistaken or I mean misinterpreted by some skin disease, and people first take the treatment for the skin disease. If there is no effect, then people go for diagnosis of this disease. Is it this a fact?
Yes, actually, I talked to some EMS people, EMS doctors, and they told me that most of the people once they have the patches, they usually come and visit to the skin doctor than neuro doctor. So they don't know, they are not aware of the fact that they think that this is a skin disease. And so they tend to visit the skin experts in EMS than neuro expert in EMS. So they should literally visit the EMS neuro experts and do the analysis work, do the clinical study, whereas they should not visit the skin experts. So skin experts refer them to the neuro. So this is usual trend, and most of the time, as you say, like patches. So usually the threshold that we use is 6 patches in your body are actually positive NF1. So if you have a patches less, if someone has a patches less than 5 or 4 or 6, then we certainly do not classify them as NF1 positive, but they are the carrier, more likely. So these are things that we are implementing in our algorithm, but hopefully once it is ready, I will certainly make it public so that everyone can use it and classify the patches. Thank you.
Anyone else having any questions? Okay. Thank you so much, sir.
Yeah, thank you.
Thank you.
Thank you,
Dr.
everyone. Thank you, everyone.
Dr. Prachi. She's there. Yeah. You are muted. Prachi, you are muted.
Prachi, your voice is you're not audible.
Some issue. Rachna ma'am, I think we may close the session.
Okay, so we can
There is some
close.
issue.
Some problem with Prachi. So thank you so much, Dr. Jiten, for your nice and valuable talk.
Thank you, everyone. Thank you.
Hello, am I audible?
Hello,
Yes,
yes,
you
yes,
are.
now you
Yes,
are
you
audible.
are.
Actually, there are certain questions which is to us. I don't know because of the technical glitch what was the reason. Like me, they were also not getting their voice. So I
Okay, so
think
they can type in chat box and we will read it out.
ये just Mr. Reyes had sent me the Wise. Mr. Wise had sent me the message. So he is asking, yes.
Hello, am I audible?
Yeah,
Yes, Vais.
go ahead. Yeah.
Hello, am I audible?
Yes,
Yeah, go
Vais,
ahead.
you are audible, beta.
Audible.
So my question was, what is the difference in the severity of neurofibromatosis between patients with and without NF1 microdeletion?
So thank you for these questions. The mostly once someone has a macro deletion, especially the deleted fragment, then they're gonna have a severe cases. Also, one interesting thing that the software is going to predict that the severity level as well. So technically, right now, when someone has an NF1, we certainly say, okay, be careful. You're gonna have many problems in future, but we cannot certainly tell the severity level. Whereas we are trying our best to implement an algorithm in our algorithm so that we can predict the severity level of the disease as well. So we notice that most of the time when some micro deletion happen in the genes, then there is severe problems happens. Okay, so yeah, thank you.
Okay, thank you so much, sir, for answering my question.
Madam, you can confirm is any other question? If so, then we can proceed.
So participants, do you have any question? Actually, no, Prachi, ma'am. We have already asked once.
You can end the session by your remarks.
Okay.
Thank you so much.
Yep.
Yes, Jiten, sir. You want to say something?
No, thank you for inviting me
Thank you.
to this.
Thank you so much, Jiten, sir. It was my really nice, valuable session. I hope our students must have learned a lot and they will try. And we all are waiting for your results just so that we can also see that which algorithm is giving you the best result.
Yeah.
Okay.
Sure.
Thank you. All the best for your research.
Thank you, Dr. Jiten Narayan, for
Thank you.
accepting our
Thank you.
invitation.
Thank you so much.
Thank you.
Thank you, Prachi ma'am. Thank you, Rachna ma'am. Thank you.
So, on the behalf of this organizing team, we have one more speaker. We are trying to connect him, Dr. Ilyas
So
Rashid.
Ilyas is online or not?
Madam, actually, I don't know what problem is.
Because
he's not
here in this total.
Just give me 1 or 2 minutes here.
Okay. You can contact individually.
Because he is currently in Dubai or somewhere outside. So I just connecting with him. Just a minute.
Okay.
Yesterday he had some medical emergency. I think for this
Okay,
reason, he just
so you can just connect. Meanwhile, we can start with the oral presentation of the students.
Unfortunately, due to some emergency, Dr. Ilyas Rashid cannot join us today. We highly regret the inconvenience caused. Thank you.
Sure. Dr. Swapnil, are you here? Dr. Swapnil, chair session को बुलाओ. Dr.
Yes, Prachi.
Madam, because of certain reason, Dr. Ilyas
Okay,
may present later on. So till then, we have our expert, Dr. Swapnil. So
Okay,
Dr.
okay.
Swapnil,
Okay, so Dr.
Just a minute.
Swapnil, you can start with your talk. Have we the topic of Dr. Swapnil?
Yes, sir, I'm just giving you.
request a 15 minutes break. We are sincerely sorry for the inconvenience caused. I would request you all to take a break of 15 minutes and then we'll rejoin at 12:15.
Madam, we will be rejoining on 12:15 because some reasons he will be joining at 12:15.
Dr. Swapnil.
We can have 15 minutes break
Okay.
and we can provide you coffee to our chairs.
Okay, thank you.
Thank you so much,
Okay.
ma'am. Can I have break for 15 minutes? We will
Okay.
start. हाँ
Hello. Can I on? अभी 14 मिनट हुए बस. Monica. Monica.
Dr. Swapnil, I think you have joined. And
Yeah.
hello. अब उठा ही नहीं रही फोन, बताओ हम क्या करें.
Hello.
Dr. Swapnil, can we proceed with the session?
Yeah, sure.
Dr. Swapnil,
Yes, ma'am.
Yes.
He is replying,
Hello.
yeah, sure.
Dr. Yes, with the due permission of chair, Dr. Meenakshi, can we start the session?
Sure, ma'am.
Sure, ma'am. Thank you.
Welcome, Dr. Swapnil.
So I would like to request my mic of ceremonies, please take over the session.
Invite Dr. Swapnil Srivastav, sir, currently working as a clinician and practitioner in Madhavbag Cardiac Clinic and Heart Hospital, Mumbai, India. He is having more than 15 years of experience for cardio rehabilitation and orthopedic healthcare. He is heading an physiotherapy center of Madhavbag. Currently, sir is trying to incorporate AI-based approaches to improvise healthcare. Now, I would like to request our session chairs to take over the session.
Thank you.
Dr. Swapnil, on behalf of organizers, I welcome you again. Kindly start your session.
So, ma'am, firstly, I would like to congratulate the MIT University Lucknow campus for organizing this excellent workshop. After that, I congratulate all the members of committee and thanks to everyone who give me the opportunity to speak on this topic. So, firstly, presently I am working in Madhavbag Cardiac Hospital in physiotherapy. I have total 15 years of experience in cardiac rehabilitation. So firstly, I want to ask you, what is artificial intelligence? Artificial intelligence is the science of making the machine that can think like humans. The goal of AI is to be able to do things such as recognize patterns, make decisions, and judge like humans. One AI market in India. So as per Economic Forum report, the AI का जो market है in future was very broad, very huge. So as per Economic Forum report जो बोलता है 11.7 billion market boom in 2025, and AI market in healthcare industry in worldwide in 2021, the artificial intelligence in healthcare market was worth around 11 billion US dollars worldwide. It was forecast that the global healthcare AI market would be the worth almost 188 billion US dollars by 2030. Increases at a compound
Dr.
annual
Swapnil?
22 to 2023.
Dr. Swapnil, sorry to interrupt you. Would you like to share your presentation? Because we are not
Pardon,
able
ma'am?
to. Would you like to share your presentation?
Yeah, sure. Just a minute, ma'am.
Yeah.
Sir, if you are having any problem in sharing your presentation, either we can share or you can take over session orally if you want. It is up to your discretion. Or
Ma'am, due to some technical issues, I am not able to share my presentation.
so with the due permission of chair, can Dr. Swapnil take over the session in
Yeah, ma'am, sure.
okay, in a verbal mode and in a communicative mode of okay. Thank you, Dr. Meenakshi. So, Dr. Swapnil, you may proceed with the experiences and share how the AI is contributing in the healthcare sector, because you are on the real person who can tell us about the current scenario of healthcare system. Dr.
Sure.
Swapnil? Yes.
Yeah. So what is the role of AI in cardiac rehabilitation? I am a physiotherapist and I am working in a cardiac hospital. So I want to ask you, what is the role of AI cardiac rehabilitation? Cardiovascular disease is one of the leading cause of death in modern society. There are many factors that must be into consideration when analyzing cardiovascular diseases, such as patient quality of life, general well-being, diet, lifestyle, medical costs, and many other factors. Uses of AI included detecting heart disease, treating stroke faster than enhancing diagnosis radiology capabilities. AI in cardiac rehabilitation, CRT means cardiac rehabilitation therapy program has been shown to be an effective secondary prevention method for cardiac rehabilitation. It can help reduce patient risk as well as improve their recovery and overall health outcomes. So we are monitoring wearable healthcare technologies, healthcare monitoring devices want the body to monitor the healthcare data through the uses of biosensor wearable healthcare technologies, accurately monitors pulses, fitness data, and AI wearable monitors, fitness bands, biosensors, wearable ECG monitors, wearable blood monitors like CGMS. CGMS is a continuous glucose monitoring device automatically essential your blood pressure levels or automatically essential your blood pressure level. You can see what blood glucose level is at any time. And we have also ABPM devices. ABPM is an ambulatory blood pressure monitoring device, digital blood pressure monitoring device. This device measure record 24 hours of blood pressure.
I think network.
हाँ, some network issue is there. I think he is disconnected now.
Just a minute, madam. I'm just connecting him through call. Okay.
Okay.
Dr. Meenakshi, Dr. Swapnil is in the process of connection. I am on
Okay,
call with.
It's okay.
No, he's in the process of connecting. Actually, he's from Bombay, so heavy rains are going, I think, with that reason.
I know.
So I know because he's trying his best. Dr. Meenakshi, I have received message from Dr. Swapnil on my chat. There is a heavy rains and for which network issue is going on. Madam, I'm trying
It's
again.
okay. We may reschedule his session later on.
Either we wait for 5 minutes.
We may wait also. I'm here. Don't worry.
Okay. Because I got his message in my mobile.
No, we understand. Okay,
But
I'm here. Don't worry.
Willing to present and to interact.
I know, and whatever he would tell us now that would be very good for the students also. That is the real-time case studies and how he is using the AI. Yeah.
Yeah, because I would like to correlate as by his CV that Dr. Swapnil is doing cardio rehabilitation and orthopedic care in Madhavbagh, Bombay Center. And that is a center where many of the celebrities are also coming. And I have majority of photographs that I can even show to the audience. And basically, previously, they were dealing with the classical concept of physiotherapy, classical concept of cardiac issues, cardiac rehabilitations. But in the current time, they have made a database based on the stages of therapy and based on the stages of cardio rehabilitation.
Mhm.
And they are trying to applying the data generation and integrating the AI approaches in the current time also. So it is one of the very leading group of Bombay, and I think it is somewhere in North also.
Hmm.
So for which I was
I
found is
have.
the right person, and because all of us are in need of the orthopedics and cardiac, we are basically students are quite young to take over, but his lecture quite be very promising for all of us. But I don't know. Bombay is one of the known region of rains, so
True.
I'm extremely apologize
Oh,
for this
no, no need. No need, Dr. Prachi.
inconvenience.
We may
Uh,
have this session later on again.
yeah.
Okay.
Uh, we can take later on session as we get the time. Or if
Yes.
he joins, I will ring you back, ma'am. Okay. Thank
Sure.
you, ma'am,
I'm
for
there. Okay.
sharing this.
Thank you. Thank
Thank
you.
you. Dr. Swapnil Srivastava, have you joined? Dr. Swapnil Srivastava, are you able to hear? इतना कुछ करना पड़ता है इसमें. Dr. Swapnil Srivastava. कुछ प्रॉब्लम आ रही है.
मीनाक्षी आ तो गई है, ठीक है फिर मीनाक्षी है, ठीक है ठीक.
पता नहीं क्या इशू आ रहा है.
आपण डायल केलेला नंबर सध्या
कर दीजिए. अब हमारे चेयर. हाँ, डॉक्टर मीनाक्षी इज वेटिंग इन द. यस यस प्लीज. बस मीनाक्षी मैम वो कर रहे हैं, पता नहीं क्या इशू आ रहा है. पता नहीं क्या कनेक्शन है इसमें. तुम कॉल
Okay, do we have any student who would like to switch on the camera and give us some feedback on the sessions which they have attended till now? Priyanshi?
Dr. Meenakshi madam, we are Sudipture. We can float the feedback form if you permit us.
Yes, ma'am. Sure.
Okay.
And we can have some video feedbacks also.
That could be very fine. That could be very fine.
If any of our students could switch on the camera and tell us something about his experience, which session he liked the most.
Ayushi and
Priyanshi, Ayushi. We have Ruby also. Ruby, please switch on your camera.
Dr. Meenakshi, Dr. Swapnil is connected over here. He's calling me.
Fine, very good.
So Dr. Swapnil, please continue your talk.
Ma'am, you're muted.
Ayushi, madam was asking some feedback. So I'm expecting from you at least you'll give. Because since beginning you are the very.
Ayushi had asked a very nice question today also.
She is very actively participating in the conference and asking
That is
very
good.
generous and very
Relevant, yes, very relevant question.
relevant questions with the speakers. Dr. Swapnil, have you, Dr. Swapnil, Swapnil Srivastava is here. Dr. Swapnil, please open your camera and unmute yourself. Dr. Swapnil, please open your camera and unmute yourself.
I think there is network issue. He is trying very hard. Ma'am, here
Yes,
Ayushi.
Hi Aishi, how are you?
I'm fine, ma'am. How are you, ma'am?
I'm very fine, and just as chair has asked you something, you can speak out the chair.
Yes, Ayushi would like to hear some feedback from you.
Okay. First of all, I'd like to thank you, Meenakshi ma'am, and also I'd like to thank Prachi ma'am for both of yours appreciation. And I'm really blessed to have a mentor or faculty like Prachi ma'am and you and other faculties of Amity University. I've been an alumni of Amity Institute of Biotechnology, whose conference I'm attending right now. And about the conference, I would like to say that today's conference I found very, very interesting. Even yesterday's information and lectures and presentation, those were also very interesting. But today's lecture I found to be very, 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 very interesting because brain and neuroscience are also one of those disciplines in which I'm like very interested. So I'm very interested in these disciplines, neuroscience and all, and medical science also. So basically this international conference is like connecting the neuroscience, medical science with the artificial intelligence and new technologies. So I'm finding it to be very, 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 very interesting. And to be very honest, it will be a memorable international conference of my life. And I want to describe about this conference in lots and lots of words, but I feel that I'm quite at loss for words. So I just can say outstanding international conference is and is going on.
Thank you, Ayushi. Thank you so much for this delightful words. And basically, if students get benefited, our motto get solved. है ना? That is the main issue. And so much, really, students like you are highly needed. Those who show enthusiastic participation and inquiries. So thank you so much, Ayushi Beta, and my very good wishes to you. I think Dr. Swapnil has joined and he's speaking out, I think. But still he's... Dr. Swapnil, can you hear me?
Yeah.
Yeah, thank God. Thank God, at least rains have permitted you to join with us. Thank you, Dr. Swapnil. Please carry on with your session.
Sure. So I will start again. So examples of AI is artificial robotics, Google Maps, maps and navigations, Google Assistant, self-driving cars like Tesla, and in healthcare industry. So role of AI in healthcare, benefits of AI for healthcare. The following benefits of AI healthcare is high quality patient lives, high accuracy of treatment, reduces cost, higher employee engagement with patients,
Sir,
streamline
I'm sorry to
workflow.
interrupt you. Sir, I'm sorry to interrupt you. Sir, are you willing to present your PPTs or you will be in the interactive mode?
No, I'm it's not possible to. I'm
Okay, okay,
unable
okay,
to.
okay, okay. So for you are not
I'm,
opening your camera because of the net issues, I think so.
yeah.
Okay, no issue. Because once you
I'm
can.
unable to share. I'm unable to share my PPT due to some technical issues and network problem.
Okay, okay. And
I will
if
share later.
you can open your camera if suits. Thank you. You may please proceed.
Due to heavy rainfall, network is very poor.
You please proceed. You just bless us with your knowledge through interactive session, please.
Yeah, sure. So benefits of AI in healthcare industry: high quality patients' life, high accuracy of treatment. It reduces cost, higher employment engagement with patients, streamlined workflows, high employee efficiency, decreased labor demand, reduces workload, and enhance protection against. So now let's a few example of artificial intelligence in healthcare system. The first is accurate cancer diagnosis. PATH AI offers one of the best machine learning and artificial intelligence tools in healthcare that allows pathologists to make accurate diagnosis. So PATH AI operates the largest independent anatomic pathology laboratory in the United States called PATH AI Diagnostic Processing. More than 5 lakh cases a year across in healthcare uses cases. Now, let's see few examples of artificial intelligence in healthcare. Customer service chatbots. Chatbots can make it easy for users to find information by responding to questions and requests. Healthcare chatbots can use information about the patient's conditions, allergies, and insurance information to schedule appointments faster and better. Benefits of chatbot is less time spent communication to the doctor's office, less money spent on unnecessary treatment and tests, easy access to the doctors at the push of a button. So we will start again AI in cardiac rehab. So I will start again. Cardiovascular disease is one of the leading cause of death in modern society. There are many factors that must be taken into consideration when analyzing cardiovascular disease, such as patients' quality of life, general well-being, diet, lifestyle, medical cost, and many other factors. Uses for AI included detecting heart disease, stroke faster and enhancing diagnosis radiology capabilities. Example, a Mayo Clinic study applied AI techniques to a new screening tool for people with a certain type of heart problem that has no obvious symptoms. Mayo Clinic is the largest integrated non-profit medical group practice in the world. AI in cardiac rehabilitation. What is the role of artificial intelligence in cardiac rehab? Cardiac rehabilitation program has been shown to be an effective secondary prevention method. It can help to reduce patient risk as well as improve their recovery and overall health outcomes. So we have some AI used in cardiac rehabilitation. Just like wearable monitoring, wearable healthcare technology is healthcare monitoring devices worn on the body to monitor healthcare data through the use of biosensors. Wearable healthcare technology accurately monitors pulse, data, fitness, and sleep. Wearable monitoring are fitness bands, biosensors, wearable ECG monitors, wearable blood pressure monitors, in
Yes, Dr. Swapnil? Your voice is not coming. Hello, Dr. Swapnil.
I think network issue is just.
Network issues
But
is
he's
causing
quite
it a
willing
lot.
to give his. That
He
is
is
a very.
very, yes, he is
Yes,
willing. And even I am also very much willing to listen that how in real world he is handling those cases.
hello.
Yes, Dr. Swapnil.
Am I audible, ma'am?
Yes,
Dr.
you are.
Swapnil. Dr. Swapnil, I think you are having heavy issue of technical. Dr. Swapnil, I think you are having a great issue of internet, Dr. Swapnil. But I
Yeah.
really salute you. Hello, hello, hello.
You may conclude, Prachi, ma'am. He is not able to listen also.
Because heavy rains, I'm just going through the
Concluding
messages
remarks.
of Dr. Swapnil consistently. I'm
Huh.
receiving his messages, Madam. I'm
I
so
know,
sorry.
I know.
You can't do anything when man proposes, God disposes. You can't do anything. So anyway, I salute
Absolutely.
your zeal of presentation, and you tried to club the session with the real approaches which you are applying for cardio and orthopedic patients. And I think Dr. Meenakshi is more competent than me, can give the brief of the session.
So we
How
actually the people who are working in the field of IT now, especially in AI, machine learning, they do need to understand that how they diagnose the problem in real sense. And then only we all will be able to have a model, we all will be able to develop some tools that can simulate their working. So that is the main reason that why such kind of people should interact with our students so that our students can understand that what are the prominent topics which they can take in as their projects or their studies. Like we all know that our students go for the NTCC. So if they understand, they can have they can make the basic tools through which they can uh, they can extract the features and they can make system intelligent and the system could diagnose in the similar manner the way the Dr. Swapnil is doing. So it's our hard luck that we are not able to hear him properly. I request you personally to please reschedule his lecture later on. And I
and
thank
I
Ayushi
will
also. Yes, ma'am.
and I will request from Dr. Swapnil. Recently, I got his message, Madam. I am really very sorry because of the very heavy rains. I am unable to, even I am not able to listen. You people, I can some
Uh,
disconnected again, Madam. Uh, but anytime I will, it will be my pleasure to deliver talk during the next time or. So I will request Dr. Swapnil whenever he will be present in Lucknow in person, we can manage one lecture for him. Isn't so, Dr. Meenakshi?
yes,
Jointly, we can propose a very good lecture
definitely,
with the
definitely.
practical things so that we can
Yes.
take care of our knees and heart.
Bilkul. We can have a workshop also, not only
Yeah,
lecture, and so they can students will learn.
so that we
And
can.
thank you, Prachi ma'am, and thank you, Ayushi. Your feedback was really very nice. Very nice feedback was given by Ayushi. Yes, Prekshi, over to you.
Ananya.
No, I would like to hear something from Prekshi also.
Oh,
Prekshi.
great.
So when you are going to tell us about the tools, huh? about the tool which Siddhin sir was saying in the morning.
Yes, you're audible.
Thank you so much, ma'am. I really like the session that happened today. And artificial intelligence, I really have to learn from you, ma'am. I don't know about it currently. I'm just a beginner. But I hope we can collaborate and we can do something and we can teach hard. Because that is not my domain.
Yes, Ananya.
Thank you so much, ma'am. I highly regret all the inconvenience that was caused today. I would also sincerely express my gratitude to Dr. Swapnil Srivastava for making time to connect with us today on such short notice. I would like to express my sincere gratitude to Dr. Rashna Chaturvedi ma'am and Dr. Meenakshi Srivastava ma'am for their efforts and contributions in running this session smoothly.
Ananya, please announce the rejoining about rejoining of session.
Sure, ma'am. Thank you all for being so attentive and patient with us. I would now request all of the participants to fill in the feedback form for this session, the link for which is available in the chat of the meeting. We would now pause over here for a lunch break. I request all of you to gather back here at 1:30 p.m. to listen to the young minds as they present their research work. Thank you.
Thank you, Ananya. So participant, please take over your lunch. I can just provide you the e-lunch. Okay. But session chair, your coffee is due on me.
Yeah.
So thank you so much, madam, for sparing your time.
Thank
And
you.
it is my very humble request. Please be present during the valedictory if you can manage.
Sure. I will be.
Okay.
Thank you.
Thank
Bye.
you so much.
Take care.
Thank
Bye.
you so much.
Sadaf bhi aa gaye.
So hello everyone. Once again, I welcome you all in the fourth session of the day 2 conference. So I request to Ananya, please proceed further.
Yes, ma'am. We welcome you all back. Thank you for gathering back on time. We would now take the honor to introduce our esteemed judges for the oral session, Dr. Aditi Singh ma'am, Dr. Gurjeet Kaur ma'am, and Dr. Manish Trivedi sir. I would express my sincere thanks to them for making time to add value to our conference and accepting the invitation to judge the session. Without any further delay, we would now like to begin our presentation session. I request all the participants to be attentive and ready with their presentation. I would also kindly remind the participants that the allotted time for presentation is 5 + 2 minutes, where 5 minutes is for presentation and 2 minutes is for question answer session. Participants are requested to restrict themselves to the time limit. I would now request our first participant, Mr. Sahastru Sinha from Amity University, to proceed with the presentation.
Thank you, Sanjana, for taking over the session. And I request, please be on the time, Sanjana and Ananya, and so that a time limit can be given to the judges. I request to give our introduction about our judges.
Hello, am I audible?
I hope all of my judges are here on the podium. Dr. Aditi Singh ma'am.
Yes, ma'am. Yes, ma'am. Good afternoon. I am here.
I think.
Yes, ma'am. I'm very much here.
I am. Aditi ma'am is also here. Dr. Manish and Dr. Gurjeet. And I hope all of you have received the judgment sheets, hard as well as the soft copies.
Yeah,
Yes.
Okay. Thank you, ma'am. Thank you so much for your precious time. Thank you so much. And I would like to congratulate Aditi ma'am for great achievement by under her mentorship.
Thank you, ma'am. Thank you so much.
Thank you so much, ma'am, for sparing
We
our
will
time.
begin with the presentations. Very kind of you.
I request first presenter, please start the session.
So, Mr. Sahastru Sinha, you can proceed with the presentation.
Yeah. Am I audible?
Yes, you're audible.
Okay. May I present my screen?
Yes, you may proceed.
Okay. Uh, the screen sharing is not optionable in my.
Just give us a second, we'll look into it.
Just I'm requesting my co-host to give you the.
Okay.
Just a minute. Just a minute.
You may please check now. I think it's sharing now.
Yeah.
I hope now you can share your screen, Mr.
Can you see the
I'm able to see
presentation
your
mode?
screen.
Presentation mode, the slideshow mode?
Yes, it is visible in slideshow mode.
Okay.
Yeah.
So, good afternoon, each and everyone. So our topic is predicting, analyzing, and managing stress in the society using information technology. So to introduce with the topic, I would like to welcome Arpita Yadav. And ongoing will be continued by Ananya, Akrama Javed, and myself to some of the whole presentation. So here, Arpita. Here you go.
Hello, everyone. The objective of our study is to utilize the information technology to develop such tools which can accurately predict the stress among the society that can that can predict the level of individuals, analyze contribution factors, and provide effective management techniques. Coming to the next slide, what are the causes of stress in society? Basically, there are so many causes like work pressure, health concerns, financial burden, and relationship issues. The next slide is about the impact of stress on society. Basically, the stress is impacting so much on our on mental and physical health, productivity, performance, and interpersonal relationships. The current method which we are using to manage the stress is basically self-reporting, psychological measurements, and psychology assessment. There are but the reason why we cannot going through this methods because we have some limitations in the current methods that are subjectively cost and accessibility, single data source. Because if we are going for a counseling and all the things, there will be so cost and accessibility is not among all of them. So there are some limitations, and for that scenario, we are using information technology to overcome these limitations. So now I would like to give my slide to Akrama to present the next slide. Thank you so much.
Hello. My name is Mohammed Akrama Javed. I'm gonna continue the slide that is given by my friends Sahas and Arpita. To inform that role of information technologies is stress management. We have done 3 portions: data collection, data analysis, and personalized interview. In data collection, we have taken data from Google, Reddit data about the people and presentation according to it. And then we have done analysis according to it with the help of the algorithms and patterns, correlation and triggering. Then we have personalized interview, and moving to the next presentation. Application of data analysis and stress management. We have divided it into 3 portions. Okay. I didn't heard you. Should I continue?
Please go ahead.
The slide is application of data analysis and stress management. We have divided it into 3 portions through which we can predict the suicidal rates and everything. According to it, the first one is early warning system. In this, there are certain words that we have decided to analyze when we check the data. It's like some specific words that I need help, and according to that, there are some specific words according to which we have set the algorithm. And the second one is virtual reality therapy. In this, data tries virtual reality experience or simulation programming. environment that helps to relax and anxiety in a controlled and immersive manner. Then there is a smart phone application that will be explained by my friend Sastanshu, and I will moving on the second slide. Feasibility study in this, there are three portions: data availability, technology integration, and ethical consideration. Access of availability in the relevant data source that we have acquired through Kaggle and Reddit. The main portion of Kaggle that we have picked up the data from and do the our analysis and everything on that. The second is technology integration, as through we have not meant to go through many other social media websites. We have moved through the social like Reddit only. We does not move to multiple social media exercises. Then we go. Methodology contains three: data collection, data analysis, and
Sorry to
algorithm
interrupt,
development.
but please wind up quickly.
Okay, my friend will continue, Ananya.
Hello everyone, am I audible?
Yeah, you are audible. Please proceed.
Okay. So here we are showing the observations and results. The first chart is having two columns where the whole sample is presented. There are some non-stressed people and some stressed people. The second chart is representing the main reasons behind people getting stressed and not stressed. The third slide is the visual representation, or you can say the pictorial representation. These are basically the word clouds. The bolder the words are, they are showing the higher the frequency. This bar chart is showing the comparison between the words distribution in texts between stressed people and non-stressed people. That it's a unigram analysis, which is showing that the stressed people are using the words like the most. This is a diagram analysis of the two most frequent words came in most of the subreddit posts by the stressed people. This is a trigram analysis basically, which is showing the frequency of words used by the stressed people. And finally, this is the count of words distribution in the text of stressed versus non-stressed statements. The rest will be continued by Sahasraj. Thank you.
Hello, am I audible? Hello. Yeah.
Yes, you are audible. Yes, you are audible.
Audible,
Yeah.
please continue.
Thank you, ma'am. Yeah. So, first of all, let me sum up what my colleague just said. So, basically, what we are doing is we are using the NLP technology, natural language processing technology, to just find out what are the major reasons of stress in the youth. First of all, we are taking the age group 13 to 20 from Reddit. First of all, because Reddit does not allow people under 13 years of age. Secondly, this particular age range which we have taken, this is because like post-COVID, these age range were the most highest affected by the COVID. So the stress level in these age range, because definitely we are accepting that people beyond age 20 have higher stress rate, but post-COVID, the most peculiar behavior was shown, like in these age range, stress just exponentially rose up. So what was the reason behind it? We tried to find through the natural language processing that we need help, or the trigraph range, or the unigraph range, or the bigraph range, which we have just shown. So it just shows like I feel like crap, or just like the PTSD or anything like that. So what we have done, we are trying to predict what the younger generation is feeling through. And the benefits is like we are able to assess, like majority of the people are suffering from it. Accessibility is like, because social media is very accessible to each and everyone. So any post which you are sharing or something like that, we are able to detect it. Cost effectiveness, because the social media platforms like provide data in a very cheap rate, so it can very powerfully affect us. And after, if we have done with the technology based stress assessment, it came, if it dealt with properly, it can empower the person who has been feeling stressed or something like that. So limitation is basically the data variability, because there are lots and lots of data, and particularly which person is feeling what, it is very difficult to accurately predict what he or she is feeling. User adaptation, ensuring widespread acceptance and usage of stress management technology among the general population. Yeah, exactly. It is very hard to ensure that each and everybody is using that kind of stress management technology. Okay. And obviously, anything needs refinement and enhancement. So future outlook is like, this is basically the prediction. We are analyzing and predicting, but how we are able to manage it. So first is like implementing a stoichiometric process to psychological tests for those persons who are feeling stressed or not feeling stressed, by an app which use some kind of some advanced sensing technologies, which emotion technique detecting variables like your heart rate monitors or the smart watches, something like that. Artificial intelligence, the use of deep learning techniques. The accuracy percentage is like 65 to 70% in this probability. So to use or to more accurately predict the scenario, we can use the deep learning techniques, which increase the enhanceability, the accuracy of the algorithm to 99%. And using of integrated system like healthcare applications with, I guess, a consultant of with the consultant or a the therapist. Okay, I guess the time has exceeded. So if you have any questions.
Are you done with?
Yeah, because the time limit exceeded, so I didn't want to continue.
Okay. So are we open for question answer session? Organizers?
Yes, madam, please.
Okay. Okay, ma'am. So Sister Anshu, as many of you have presented, first I would like to know who's going to answer the questions. I think in presentation, in oral presentation especially, there should be only one presenter. Then only we are going to, judge according to the presenting skill or likewise. I think that is one question I have to ask
Very
the
true, madam. Even I was
organizers.
also not knowing these three are going simultaneously to present.
Yes.
Actually, the paper was like in a combined effort, and it was a teamwork. So we thought that each of us should present. I'm really sorry for that. But I would be able to answer your questions if
Anyway,
how much as
okay.
possible.
Okay. So one question from my side, and then there are other judges also. I want to know that how you have decided stressed people and non-stressed people, because all your data is whatever the graphics you have shown, they are divided into two groups: the stressed people and non-stressed people. So
Yeah.
how you have done that right from the start? What was the criteria?
Ma'am, the criteria was like that. The training phase, we have kept some sort of phrases like I don't feel so good, I need help, I am stressful, in a very stressful situation. There are phases in that. So with the help of
Are these clinically proven criteria to categorize anyone as a stressed person? Have you gone through any
there are
literature?
some clinical terms like I'm feeling through post-traumatic stress disorder. So these are the clinical terms which we have taken, but the terms which normal the non professional people use. Like I feel like not so. I don't feel so good. I'm feeling depressed. I'm feeling anxiety, anxious, or some lot like that. So first of all, which we have taken from the clinical phase, it is like the with the help of some friend of ours, which is doing psychology in the Amity University. But after that, most of us, it's like the data which had been kept in Kaggle, it categorized like it is stressful. These words are stressful and these are not stressful.
Okay,
So
fine.
it was
Okay, fine. We'll go for other questions also because we don't have much time.
Okay, yeah. So Ranchu, I have one question for you.
Yes.
Because when it comes to, dealing with the social media, how true you people are when it comes to, accessing the or using internet or any social media.
Yeah.
You
but
can't be
if
true
someone is
self,
posting on
right?
social media about their concerns that
Please.
we are feeling not so good, because anything to express yourself in a social environment or in public, it's first the most difficult thing to deal with. Okay, after that, if they're accepting that I'm not feeling so good, I'm suffering from stress, so that means that it is mostly accurate.
Well, I may have a different opinion on that, but
Sure.
it's okay. Thank you. That's all from my side.
Actually, I joined a little bit late, so I could not heard much. So I just evaluated him on the basis of the explanation on questions asked by Dr. Aditi and Dr.
Okay, okay. Okay, thank you. We can go to the next one. Thank you, Sister Anshu.
Thank you, ma'am. Thank you so much.
Our next presenter is Mihir Tandon from Amity University.
Good morning and hello everyone. So I would like to first ask one question because we have a group PPT. So should I be sharing alone and am I allowed to do with my colleagues?
Presentation should be done by just one person, whether it is a group effort or whatever.
It's totally all right.
I think
I'll be sharing.
all the other judges will also agree to that. So
It's totally I'll be sharing for
Absolutely
alone. It's
right.
totally all right. Just a minute, I'm sharing. For some reason, I'm unable to share my document here. I don't know why.
Just a minute, I'm taking care, Mihir.
For sure.
प्रेजेंट नहीं कर पा रहे हैं, आप उनको अलाउ कर दीजिए. Beta, host has already permitted you all.
I think just a second. I'm trying it once again. I'm going to refresh it.
It may be any technical glitch from your side.
For sure.
Because host has already given all the rights.
Yeah. Can my friend share the PPT because I'm unable to share it?
So you can.
I'm gonna explain it. But
that's fine, I suppose.
okay, just okay. I'm telling her.
I would like to request from all participants, it's better you give your presentation by using laptop, because sometimes from mobile and this interface may create any issue. So it is requested to better use your laptops for presentation.
Totally, ma'am. Just give me a second, ma'am, please.
Sure.
Uh,
Sanjana, you can move forward for the next presenter. Till then, Mihir may.
okay, ma'am, I'm fixing the problem.
Till then, you can
Okay, ma'am.
resolve your issue. Okay.
For sure, ma'am.
Our next presenter is Akshat Sinha from Amity University.
Ma'am, am I audible?
Yeah, you are audible.
Okay, ma'am. So good afternoon all. This is Akshat from Amity University. So my topic of the presentation is role of artificial intelligence in the management of inflammatory bowel disease. So first of all, what is inflammatory bowel disease? In short, we call it as IBD. So what is IBD? IBD is a chronic gastrointestinal tract inflammation in the gastrointestinal tract, and there are mainly two forms of the IBD, that is a Crohn's disease and the ulcerative colitis. So
Akshat, your presentation is not presented. Look into that.
Presentation is not visible.
Not visible.
No. Please share your slides.
Wait, ma'am, is it visible now?
Not yet.
Shared.
I don't know what is the issue going beyond.
You can just share your screen.
Yes, ma'am, I'm just sharing, but it is not working.
It's not visible now.
Ma'am, it can't share because I'm just allowed, but it is not. Ma'am, is it visible now?
Yeah, is it visible now?
Yes, visible now.
Okay, ma'am. So my topic of presentation is role of artificial intelligence in the management of inflammatory bowel disease. So what is inflammatory bowel disease? Inflammatory bowel disease is a chronic disorder of the gastrointestinal tract, and which manifests in recurring gastrointestinal inflammation. So there are basically two types of the inflammatory bowel disease, that is the Crohn's disease and the ulcerative colitis. So the basic difference between the Crohn's disease and the ulcerative colitis is on the basis of the location. That is, in a Crohn's disease, it affects from the GI tract. It affects your GI, any part of your GI tract, that is from the mouth to the anus. While in an ulcerative colitis, it affects only your rectum region and the colon region. So importance of IBD management. So why it is important to manage the IBD? So by managing the IBD, we can improve the quality of a life of a patient. So by by reducing the symptoms and the severity, we can improve the quality of a life of a patient. Symptoms control: by suppressing the symptoms like abdominal pain, diarrhea, and fatigue to the patients, we can improve the quality. Preventing complications. During IBD, there are certain complications takes place, like there's a development of structures, fistulas, abscesses. So by managing the IBD, we can prevent these complications. Reducing hospitalization. as in a IBD, there are the chances of the complications or there's the chances of the surgery. So by managing the IBD at the initial level, we can reduce the hospitalizations of the patients. Research and innovations, by acting on a new target, by developing, by finding a new target, we can develop a new therapies and a new treatment strategies. Artificial intelligence in IBD, how it they both are correlated. Actually, in artificial intelligence, it includes computer systems and the algorithms, and they are help to diagnosis, treatment, monitoring, and the management of the IBD. AI systems includes machine learning, natural language processing, and data analysis process. So they are help in interpretation of the patient data, make predictions of what that is the adverse outcomes, and assist healthcare professionals in making informed decisions regarding the care of the IBD patients. So need of AI. Why there is a need of AI in IBD? Actually, what happens, the endoscopy which is performed by the physician or by the operator, the quality of the endoscopy is depend upon the operator. So there are some pathology limitations also, like operator expertise, inter-observer variations, inadequate to indicate the degree of mucosal healing. So how AI work in this? So by the help of AI, we can distinguish that there is a ulcerative colitis or the Crohn's disease, or the histopathology is done by the AI. So there are the AI applications like pathogenesis, diagnostics, treatment response, prognostication, care delivery, and training. So there are the applications of AI in IBD. From the diagnosis to the prognosis, AI is involved in the IBD. Like how? In the diagnosis, we can distinguish the disease and prediction of a high risk of patients group and prediction of the IBD development. Predicting disease course and response treatment, it helps in the treatment by supporting the clinical trials. How? Standardization of a disease assessment, biomarkers discovery, and new target discovery. Role of AI in the treatment of IBD. So what are the requirements and what are the enhancements? Requirements include your data, method, and interpretability. The data, requirements in the data, that the data should be of sufficient number, representativeness, and extensive, and the coverage. The coverage should be large. The method which is adopted should be adaptation to the data type, and high accuracy method should be there. Definitely sensitive and specific method, and reasonable cost method. Interpretability, how to interpret the images which is used, visual images, and the codes which are used should be readable. Enhancement, etiology, that is a cause. So evaluating the relevance between the extraintestinal manifestation and gene and the influence of environment. How diagnosis, analyzing image by analyzing the images, enhancing non-invasive tests, determining subtype and disease status. Treatments include that is prediction, prognosis, forecasting efficacy after clinical interventions. So there are certain future visions, that are the foundation models. Foundation models, these are the AI generated models actually. So omics data, that is the genomics, transcriptomics, proteomics, and metabolomics. By getting these data, we can target our disease. So electronic medical records like histopathology, laboratory results, past diagnosis images. So AI help in all this. These are the certain references. And then thank you.
Okay. So you have told the role of AI in IBD.
Yes,
so
ma'am.
first, I would like to know: have you yourself have done any work on this, or you have just studied about it?
No, ma'am. I have not done any work on this.
Okay.
I'm just reviewing the papers.
Okay. And have you found any actual example or case study where AI has been implemented, not hypothetically, where it has been
Yes, ma'am.
shown
In
to work?
endoscopy, it works a lot as it provide a visual images, 3D pictures or the visual images. It helps to identify that there's a what is the state of the inflammation in the colon.
So how AI has been integrated into it? Can you just elaborate on that a bit?
Ma'am, as there are certain softwares or there are in the endoscopy, there are certain softwares where the visual pictures takes place, or AI helps, and there are certain wearable devices are also available, like mobile apps are also available. And these they help in real time. They help in collecting the real time data.
Okay, fine. And lastly, what are the limitations of AI? If you are talking about, is it always only the good things, or is there any limitations also?
Ma'am, there are certain limitations like what we can say that the job opportunities for the operators that perform the endoscopy and all. AI can minimize the jobs in that area also.
That's all from my side.
Thank you, ma'am.
Yeah, so Dr. Manish, would you like to continue? Or?
Okay. Actually, basically, you have given a kind of information about the AI and its involvement in the IBD management. So, would you tell me how what are the basic parameters that can be included in order to generate model for the AI? If you are working with any particular disease to implement the AI, you have to actually consider certain parameters.
Sorry, sir, I'm not able to listen you. Your voice is fluctuating actually.
It is a bit breaking, sir. Manish sir, your voice is breaking.
Sure. Okay. Can you hear me? Anyone?
Yes, sir.
Can you hear me, Akshay? No?
Yes, sir. Yes, sir.
Yeah, I'm just asking that what could be the parameters here in order to generate the model for AI, in order to manage the IBD?
So what are the parameters? Sorry, what are the parameters?
What are parameters?
Yes, sir.
You will consider to generate the AI to manage the IBD, Crohn's disease.
Okay, what are the parameters to be generated?
To be used.
To be used. Yes, sir. Like there are real-time data, as I said, it should be implemented, and the endoscopy should be performed by the AI. It should be implemented. So these are the certain parameters that should be implemented so that we can.
Performed by the AI. AI can utilize the endoscopy data.
So yes, it should be utilized so that we can get a real-time data or the exact state of the disease.
Because maybe you must have referred number of papers, right, in this regard. So
Yes,
did you find anything like what could be the status in India on this, like the involvement of this AI in IBD management?
No, sir. Sorry, sir.
You have any information on it, that if any institution, any hospital, any particular organization or any institution using such type of approach?
No, sir. Sorry.
You didn't find anything like that,
no,
right?
Sir. Yes, sir.
Okay, actually.
Thank you, sir.
That's all from me.
Okay, thank you. We can move to next one.
Yeah, I think we should. Yeah.
Our next presenter for the day is Netisha Som from Amity University.
Good afternoon, everyone. My name is Nitisha. Should I share my slide?
Yes, go ahead and stick to time.
Okay, ma'am. Good afternoon, everyone. Am I audible?
Yeah, you are audible.
Okay, and ma'am, is the presentation is visible?
Yeah, it's visible.
Okay. Thank you, ma'am. My name is Nitisha. So my topic for the presentation is predictive modeling of PCOS risk factors and diagnostic outcomes, a machine learning approach. So when I was studying about this topic, PCOS, polycystic ovary syndrome, I've learned a lot of things like
Can you please turn your phone to the landscape mode for the clarity of the presentation?
okay, ma'am. Ma'am, is it okay?
Yes, thank you.
Okay. So I've learned a lot of things from the research. September is the month of PCOS awareness, and the blue ribbon is represented, or it is a symbol of PCOS awareness. So most people don't know about what is PCOS, even most of the women. So I would like to discuss that what is PCOS. PCOS is basically a medical condition in which women ovaries produce immature or partially mature eggs in large numbers, and these large ovaries and because of that ovaries become large and secrete large amount of male hormones, which cause infertility, irregular menstrual cycle, hair loss, and abnormal weight gain in women. What are the causes? There are so many causes that can be what there are so many reasons of the because of that women are affected from PCOS. That, for example, excess insulin production or androgen production, which is androgen is mostly found in male. If it present in female, it will lead to uh, ab- um, abnormal hormones, and which cause hair growth on face and body. Uh, and PCOS can be caused because of heredity and low grade inflammation. Let's look at what are the common signs and symptoms. Uh, so when I was a kid, I used to ask my mom that, "Mama, why मैं इतना मीठा क्यों खाती हूँ?" And she's be like, "कि बेटा तुम्हारे पापा भी खाते हैं." And now I realize that my father does not have a uterus. So excessive craving is one of the most common sign in women, which can be the reason of PCOS. So the first sign is irregular menstruations or absence of menstruation. Second is heavy menstrual bleeding, and the third sign, the symptom that can be, these are the common symptoms. The third is excessive hair growth, including on back, belly, or hair loss. And fourth is dark skin, matlab skin darkening on neck or under the breast, acne. Most in teenage we all face acne, but if it's continue and if it's too much, or like on body or chest or upper back, it's an issue. And weight gain. Others, according there are so many. PCOS is not only about ovaries. If a woman is late, or if if it's too late to be diagnosed, there are other issues that can be affecting a woman's health. According to WHO, people with PCOS are most likely to have other health issues like type 2 diabetes, high blood pressure, high cholesterol, heart disease, or uterus cancer. PCOS can also cause anxiety, depression, and negative body image. Some symptoms such as infertility, obesity, and unwanted hair can lead to social stigma. This can also affect others' lives, such as family, relationship, work, and involvement in community. The things I've learned from the research are that most people are not aware, so awareness is important. PCOS is effect in 5 to 10% of females, and women between the age of 15 to 14 are mostly affected, and 70% of women are still remain undiagnosed. Sorry. Another thing that I've learned from this research paper is that uh, government should do something about this. They lack of policies, and policymakers should take a step on this. What are the treatments? There are two types of treatment. You can go to the doctor and they prescribe you medicines and all. And other thing is you can do by yourself, that is home remedies like maintaining your body weight, doing exercises, and be active, and limited carbohydrate consumptions. That's it. Thank you, ma'am.
Should I start?
Yeah, please.
Okay. So that was your just a general topic which you had taken about PCOD, is it?
Yes,
Or
ma'am.
have you taken any specific aspect of it? No.
No, ma'am. I've done only research paper right now. I've read research papers only. I'm
So,
still working on it.
So in this general, what should we ask? Well, the only thing which I can think of asking is how you can think AI can be integrated into this problem, because that is what is our major focus today for the conference. Can you think of anything?
Ma'am, it can help in diagnosis because I've read in so many papers that there are so many means the issue is that diagnosis is not easy of PCOS.
Okay.
I think so. AI can help.
Okay, ma'am, sir.
Yeah, I think I'll take a call on this because Nitisha, what I could gather was what Aditi ma'am has already said, that you just summed up on PCOS, but there was not any contribution from your end, or right? So, with regard to PCOS, do you ever you have read through the publications? Is there any remedy? Can you get rid of it?
Yes, ma'am, there are less matlab, carbohydrate consumption should be less, regular exercises, you can drink cinnamon tea, are the remedies, ma'am. These can.
Well, there's no cure for it.
No,
Nitisha,
ma'am,
let
there
me
is
be
no.
very
No,
clear.
ma'am. Yes,
Let me
ma'am. There
be
is no
very
cure,
clear on
but
this,
yes,
okay?
ma'am.
Yeah, and I think correlation or your input should have been there.
Yes, ma'am.
That's all from my side. Dr. Manish, please, you may take up.
I think okay. So just you know that what is the occurrence rate or
Sir, you are not audible.
you can hear me or not?
Yes, sir.
Okay, I'm asking like what is the occurrence rate of this PCOS in India or in or globally?
Sir, it's only 5 to 10% right now.
Global or in
So
India?
it's like if there are 5 women, one is affected by PCOS.
Global.
Yes, sir.
You mentioned two terms PCOS and PCOD. What is the difference between them?
So they're basically same, sir, actually.
What is the difference? It's basically same, right? Means there will be some difference, or is
I have no idea about it, but they are same. I don't know about the difference, but I have no
you
idea about
mentioned
it.
these two terms right there in your slide?
Yes, sir.
Okay, Nisa, that's all.
Thank you, sir.
Okay,
Yes, thank you.
Our next presenter is Mihi Tandon from Amity University.
Am I audible?
Yeah, you are audible.
So is my screen visible? Is it visible?
Yeah, it is. Please
It is
continue.
visible. Go ahead.
So my topic is applying data analytics for the study of suicidal rates in India. So I'm presenting from behalf of my group members. My group member consists of Sharon Tandon, Mohammed Farhan, and Natasha Saul. So for the introduction part is that suicide is a complex and sensitive issue that affects people of all the ages, genders, and socio-economic backgrounds. And it's one of the sensitive case because listening of an suicide cell, we just got goosebumps because it is one of the highest things that possible due to both education or some are employments and all that. So the main objective, or we can say the goal of this exploration, the factor that contribute to the suicidal behavior and recommend strategies to prevent it. We aim to understand the human side of suicide by analyzing demographic insights, regional patterns, and common suicide methods. So moving over the methodology which we have used, we have collected various data from various sources, and we have used data source from Kaggle for analyzing and using an EDA process, exploratory data analysis, which includes National Crime Records Bureau and World Health Organization. Our analysis includes variables like age, genders, occupation, education, and society methods. So we will go over to demographic insights. So one of the three top most are age disparities, gender disparities, mental health. Elderly people are more prone to suicides than aged people. Suicide shows that loneliness, poor mental health, and often prime reasons behind the suicide in the demographic. Obviously, if someone's age is higher, there are chances they are feeling lonely and they are facing a depression. So may they get into a feeling of suicide. Although that is not the solution, obviously we wanna talk about it in the coming slides. Gender disparities. Males are more likely to die by suicide than female. And we have an proof of also that because we have done an analysis on that. In the coming slides, we have some screenshots over that. With a ratio of 2:1. Societal expectation, financial pressure, and mental health stigma contributes to this trend. Now moving over to mental health, which is one of the, I think, highest factors which are happening. Issues like depression, anxiety, substance abuse increase the risk of suicide. These issues often go undetected and untreated due to limited access to the mental healthcare services. So now moving over to regional patterns. There are three basic main regional patterns which we have found, which is rural-urban divide, geographical distribution, and regional disparity. So rural area have incident of suicide than urban areas. This can be attributed to a factor like poor socioeconomic condition, limited access to healthcare and education, and a lack of awareness about mental health. Now moving over to geographical. So suicide rates vary significantly across different states in India. Southern and eastern states have higher rate than northern and western states. Kerala, the highest ranked state that had a rate of 24.5 suicides per 100,000 people. Now moving over to regional disparity. Suicides in multidimensional issue that is influenced by various region, culture, and economic factors. Analyzing regional pattern is important to understand the issue and address. So these were the three regional patterns, main regional pattern which we found. And moving over to socioeconomic impact, which are the main three impacts are occupation, education, income. Occupation. Now, moving over to occupation, we can say that it plays a significant role in suicide rates. Farming, fishing, daily wage laborers have the highest rate of suicides in India. Issues like lack of access to resources and crop failure contributes to this trend. Now moving on to education. Education is a protective factor against suicide. Literacy and limited education are often associated with a higher likelihood of suicide. Educating people about mental health can help prevent suicide. Now moving over to the income bound. Income inequality and poverty are major contributors to suicide rates. And I think we all can agree on that factor. people from lower socioeconomic backgrounds are more vulnerable to suicide due to lack of resources,
You
access to
only have
healthcare,
45
and
seconds
stress
left.
living
Please
conditions.
conclude your presentation.
For sure. So common suicide methods are like self-poisoning and hanging, which we have found. And for example, like firearm and jumping from sides are less common than self-poisoning and hanging. Now, human sides are, what we can do is we can give some empathy to the human side, humanizing the data hope. Because in empathy, we should treat with everyone with empathy, because these are some things which can prevent suicides. And for example, that humanizing the data, statics can hold and impersonal stories, experience can humanize the data and convey its impact more effectively. Now, hope always, there is always a hope. Whatever happens in your life, there is always a hope.
Please note your 5 minutes have been finished. Kindly move to the conclusion.
Okay. So the conclusion are, these are some results which we have taken out. These are the results showing the suicide rates in India during 2001-2012, females and males suicides, the 10 top 10 states, societal states versus gender, age group versus societal, all these states are available. Family problem is also available. Love affairs, mental illness, and gender versus total mental illness is also here. And these are all the exploratory data analysis we have done on the for our topic. Any Q&A, any questions?
Judges, please note participant has taken 6 minutes for the presentation.
But one thing which I just observed was in your age group wala slide, you have shown the number of suicide per state. I think that title got wrong or something. The last one which you showed. Yes. Age group versus total suicides में age group नहीं दिया हुआ है ना? वहाँ पे total number दिया है. Next में last वाली slide पे जाओ.
This age group versus total suicides happening because of family problems.
There is another one, no? Which has got only one graph. Anyway.
The graph and this one.
Okay.
And this is why the age
this
group
one
versus suicides because of hanging.
Age group versus total suicides.
Yeah,
where is the age group in this? No? Where is age group? That is what I said. It has got states versus total suicides.
Yes, ma'am, it was states. I'm really sorry. I think I mistyped it.
Okay.
It's for states. States because of hanging.
I have only one major concern, whereas I really like your topic and it's a very pertinent thing which you have worked on. But my thing is that you have only, why you have seen, taken the data only from 2001 to 12. It's been like more than 10, 11 years after that.
Yes, ma'am. Because we was just searching and we found that the most accurate data we can find on that point, and there are not the to the point data we are able to find data sets, because we have worked on data sets. We have used our Python to done the EDA.
But the conclusion which you are taking out, suppose if in 10 years the reasons are skewed, definitely there must be other reason, or you can find the age group as a different this thing. So how you can, like, very strongly conclude your findings for this? That is my only thing. Okay, we go to other questions also.
Yeah, Mihir, can you hear me?
Yes.
Yeah, I just have, can you just scroll back to the title of your presentation?
Is it this way?
Okay, you have just applied the data analytics for the study of societal rates. Yeah. So I was, wondering, when you do such kind of stat long span, you never mentioned even once about the, the psychological aspects related.
Actually, we have given 5 minutes only. Actually, the voice is cracking a little bit. I'm not able to get it properly. Excuse me, ma'am, your voice is cracking. Actually, I'm not able to get it properly.
Ma'am, your
I was
video is paused. I think there's some connection issue. I suppose. The question did not come very clearly, Pujit, ma'am.
Yeah, so now is it okay if I speak a little loud?
Yes.
Okay. Okay. Yeah, so, with your kind of analytics that you have done, anything that you can contribute or any, psychological aspects
For sure,
were there
also
taken care of?
Okay, whatever part you have heard of.
I also have some, glitches.
Yes, ma'am. Actually, voice is cracking, but I get your question. I get your question. I'm going to answer it. Ma'am, we have done a report, a full research paper on that. We have that, but we have given only 5 minutes to present that. So, concluding in a PPT of 5 minutes, but not possible because obviously there are psychological impacts in that. One of the main impacts which happen, I think, that bullying. So one of the big examples is bullying in schools, in colleges, or maybe high level of rankings. So that can impact a student mind. And there are some cases which we have found, which we were in our research paper, that there are suicides caused because of that psychological impact.
That's all. That's all. Thank you.
So much. Should I share?
Okay. Okay, I think now we may proceed for further the next one.
Thank you so much, everyone.
Our next presenter is Jisan Arif sir from CSIR IITR.
Is the presenter here?
Zishan sir, are you here?
Ananya, just make sure that your presenters are present and in line. If they're not there, then you can.
Ma'am, actually, he was in the meeting. I don't know what happened.
Okay.
Then you may call other one who are present here.
Moving to the next presentee, we have Mohammad Tana from Amity University. Sir, you may proceed with the presentation. Mohammad Tana, are you there?
Hello. Yes. Ma'am. Hello.
Yes, Mohammad Tala, we can hear you. Can you start your presentation, please?
Yes, ma'am. Give me a moment, ma'am. I'm just connecting.
In the meantime, Ananya, you can call for the next presenter. Whosoever is present may write his or her name in the chat box. It'll be convenient.
Actually, we have informed them.
Okay, that's fine. Yeah.
Ma'am, we have Mohammad Tala with us. Sir, please continue.
Share your content and unmute yourself.
Hello. Hello. Ma'am, am I audible?
Yes.
So the topic of my research paper is utilizing machine learning for the cardiac health prediction. So starting with the introduction, I would first like to introduce what is machine learning. Basically, machine learning is a way to predict information from the raw data that we have, and we could use that impactful information for our research papers and for the projects. So basically, the machine learning has a diverse field, and the scope and implementation has been increasing day by day. So here, as we all know that the cardiovascular diseases are very common these days, as I described the ranges of conditions that could affect our
Please,
health.
presentation on slideshow mode. Thank you.
Okay. The primary reason of the death is, I think, is because of cardiovascular diseases right now, and our project is also helping us to predict the people who can diagnose with the heart disease based on their medical history and the data that we will taken from the Kegel. So here the project mainly focus on 4 data mining techniques. That is, number 1 is a support vector machine, is KNN classifier, decision tree, and random forest classifier. After analyzing all these 4 techniques, we have found that the best accuracy of the data mining is of the KNN classifier, that is 87%, and we are using the data mining techniques that increase the accuracy and the efficiency of the model. The objective of this project is to check whether the patient is likely to be diagnosed with a cardiovascular disease based on their medical attributes such as gender, age, chest pain, and the fasting sugar level. And these are some of the key attributes that results in the cardiovascular diseases. By using the data sets, we can predict whether the patient can have an heart disease or not. Here in this project, I have used the 14 attributes of the patients to classify that if a patient is likely to have an heart disease or not. These are some of the literature surveys of the previous authors who have been studying on the same procedures, Mr. Sanathan Krishna and Dr. Geeta, who have in their research paper has tell us about how the patient occurring heart diseases in the patients in terms of the percentage. Here, the model that she has used is about the decision tree and the naive Bayes, and the accuracy of the decision tree is greater. And in the next survey is of Mr. Marimuthu S. Devrani and Gayatri R, who have been analyzing the heart disease prediction by using the machine learning techniques to achieve or to make the system more efficient to predict the chances of the heart attack. She has used about the KNN, naive Bayes, and decision tree and support vector machines. The data collection and the methodology. So the methodology is basically the steps that we are going to proceed with to get the recognized data patterns for the knowledge of the user. In the first step of our methodology, we have done with the data collection. We have taken the data set from the Kegel. In the second stage, we have abstracted the significant values. And in the third stage, we have done the data processing, which deal with the missing values or the redundant data or the data that is not useful for us in predicting the heart disease. So after the pre-processing, we have used the classifiers that are K-neighbors classifier, support vector classifier, decision tree classifier, and random forest classifier to handle those, to give those data as an input to this classifier and to generate the output whether the person can have a heart disease or not. Finally, we have proposed the model is undertaken and we have evaluated our model on the basis of the accuracy and the performance using the performance metrics. The effective heart disease prediction model has been developed using different classification models using 13 medical parameters such as chest pain, fasting sugar I have mentioned before also. Now in the next slide, this is the data set information. We have the age, sex, the chest pain, the resting blood pressure, cholesterol, fasting, blood sugar, and etc. They are the some of the attributes. And here in this graph, we are seeing that the target variables, there is a slightly imbalance between the number of persons who don't have a heart attack and the number of persons who are having the heart attacks. So this graph is basically telling us about the ratio of the class imbalance. The steps that we are following here is the collecting the heart disease data set, then the significant values extracted, then we have done the data preprocessing, and then we have done the splitting of data. We have divided the data into two sets, that is the training data set and the testing data set. So for the training data set, we will use the machine learning algorithm, and we will train our model on the basis of those data, and then we will test on the testing data to predict whether the person can have a heart disease or not. These are some of the machine learning algorithms that I have used here in this my model. That is the k-neighbor classifier, support vector classifier, decision tree classifier, and the random forest classifier. These all of the 4 machine learning algorithms are the supervised machine learning algorithm.
You are requested to conclude your presentation as the time is up.
Okay. So basically the results are here. We can see that the risk of the heart attack on the basis of their ages is a graph, and there is a risk of heart attack on the basis of the resting blood pressures. In the conclusion, I would say that the model that I have created was a full model, and the KNN model was the best model out of those 4 because it has predicted 87%. Its accuracy is 87% as compared to the other models, and the accuracy of the model is 80%. Using more of the training data ensure that the higher chances the model can access and predict whether the given person has a heart disease or not. Thank you.
Okay, that was a good and required study which you are going. So it's an ongoing study, or you have completed this prediction thing?
No, ma'am, it's an ongoing study because I have to use other algorithms also to predict it, because it's a very sensitive thing, because there are many medical attributes that are missing here in this data set that I have found, because the 13 attributes cannot predict that the person can have a heart disease or not. There are many other factors also.
Yes.
Therefore, it's a very vast study, but I'm trying to starting.
That is what I was about to say, that what are your performance metrics and what are your outcome metrics, or whatever data sets you are taking, how you are finally validating whether your outcome or the prediction has been so is there anything which you will be doing that validating your outcomes or
if you talk about the machine learning thing, that I will use the confusion metrics to whether to predict that what is the error are there in the data set, and it will only just predict about that. It will not give an estimate answers. So prediction is a different than going to something like yes or no, because I have to add many medical attributes in this data set so that the model can predict.
As it is 87%, I suppose is not a very promising percentage if you are talking of the accuracy. Like we, for any wet lab thing, we go for specificity and sensitivity more than 90% or 95% likewise. So having it at 87% doesn't seem very, very promising to me, at least.
Actually, the data set I have shown the graph also. There is a light imbalance in the data set. There are many persons that have higher chances. Therefore, the model predict a little not that accurately because of class imbalance.
Okay, thank you.
Thank you.
Okay.
Mohammed Talat, so you have already told us like you are going to include some other data sets also.
Yes, sir.
Even that can validate your result, because this at this stage you cannot say what could be the significant result here, right?
Yes, sir.
What is the size of your sample here you have taken?
Sir, actually, it is a very less number. It's just 313.
So that the numbers of the sample size is also very small.
Small, yes, sir.
And once you are talking about like AI and all these approaches basically get more efficient once you have a large data set.
Yes, sir. Larger data set.
If you are considering like very small data set, again it is going to be inaccurate like further if you are going to validate like on a field or in the market or something.
Yes.
So I would say to increase your samples size to here. And what type of database you have referred to collect all these data sets?
So I have collected the data set from the Kaggle, and I've just created it a CSV file, and I have used the Jupyter notebook to implement all these algorithms.
Yeah, because before using any data server or database, you also need to
Validate it.
do the literature survey or some other things to validate, like if they're really like being used by the many researchers and all. Like we must have to be sure first if they are validated or not, right?
Yes.
So okay, so but I think you need to increase it the sample size and also increase the data set. So I think you must go for it, like all these things.
Yes, sure. Thank
Okay,
you.
good.
Thanks.
So, Mohammed Tala, actually, it's a good initiative that you have taken, especially in terms of, cardiovascular disorders. No doubt, it's really, if once it is set up, it's really going to be very helpful. But I think the other aspects that we need to keep in mind is, the other diseases which either culminate into or help to increase the cardiovascular disorders. I think they also need to be kept in mind. The data algorithms for those diseases also need to be matched with what you are trying to say, right? So there has to be some balance between them also. Is there any thought process that you have?
Ma'am, I have mentioned earlier also that I have to include many attributes, or because these chest pains and the fasting blood sugar will not accurately predict, because there are some more diseases the patient can have previously, and because of that the cardiovascular disease can happen also. As my data set is not that much large enough to predict that accurately right now, but I will work on this project to make it a more better way, so that I can take a larger data set with a larger diversity of patients of different suffering.
And also like coordination with the, different hospitals and clinicians, so that you can have concrete
valid data set, yes, sir.
that you can conclude something at least.
Yes.
Yeah. So my best wishes to you.
Thank you, ma'am.
Next presenter is Sadaf Arfi from Amity University.
Yes, ma'am. I am audible.
Yeah, you are audible.
Ma'am, is my screen visible?
Yeah, it is visible.
Okay, ma'am. Thank you. Okay. Very good afternoon to one and all present here. Myself, Sadaf Arfi. I am pursuing PhD from Amity Institute of Pharmacy, Amity University, Lucknow. Today, my topic for oral presentation is artificial intelligence, a transient tool for treating cancer. My screen is not moving. Basically, artificial intelligence is a branch of computer science which deals with mathematical algorithms to mimic the ability and to work like of human brain. AI is effectively used in various healthcare sectors, like it includes various biological abnormalities, diagnosis, treatment, clinical progression, as well as life-threatening disease like cancer. As we all know, cancer is a abnormal growth of cell in which we can't control the growth of cell. It is develop abnormally, and the cells multiply abnormally. Cancers, the treatment of cancer is generally radiotherapy, mammography, imaging, cancer progression, and AI is also used effectively in radiotherapy, mammography, imaging, cancer progression, cancer genomics, molecular signaling, pathophysiology, drug discovery, chemotherapy, immunotherapy, as well as other treatments also. AI is generally intelligence that is produced by man
and it works like that of human brain. It has the capability, like it is a combination of computer science, cybernetics, neurotherapy, physical, psychology, as well as linguistics. AI basically depends on deep learning, deep neural networking. It was originated in 1943. The deep neural networking systems are involved the input layers, which includes the nodes, which we also call as units, the data that is incorporated in form of large data sets into the input layer, then it will through various coding systems, it transfers into the computed values into the hidden layer connected to the output layer. As I've already told, a layer comprises of set of nodes called units or features, which connects the input layer to the output layer for data transformation. AI is basically divided into deep neural networking is basically divided into 4 types: conventional neural networking, fully conventional neural networking, recurrent neural networking, and generative adversarial networking. It is pictorial representation of how AI is using in cancer. Artificial intelligence is using in cancer imaging. It is using in drug discovery and development, in determining the targets where we can give the drugs, and it is how to be more effective to that target sites. It is also used in treatment of cancer patient, is monitoring the patients, and used in radiotherapy. It is used in chemotherapy as well. Then how it is used in radiotherapy? Radiotherapy is generally a treatment technique for cancer patients. While using radiotherapy, while using AI in radiotherapy, it acts as a tool for enhancing the quality, standardization, and speeding up the result that will provide the patients in safe and effective radiotherapy. AI-based models, there are various AI-based models which is available in the market that ensures that how patient can achieve or can how it overcome the disease by using a well-planned treatment, so that it will not provide any side effect to the patients. As we all know that cancer therapies which are available in the medical healthcare system has various side effects to the patients. Like while using AI in radiotherapy, there are the models or the AI-based systems which are available in the market works through these flowchart. Firstly, ensure the objective, then we involve all the data sets, then data characterization, then we bifurcate according to the patients that at what stage the cancer is progressing. Then we implement our data set and the properties which are getting from the patient, then we'll ensure and ultimately will fit into the model and provide the radiotherapy treatment. AI in cancer imaging also it is using efficiently. The implementation of pattern identification in the field of medical issues has been put forward in early.
Requested to conclude the presentation as only 1 minute is left.
It is also used in cancer imaging also. Cancer imaging, we can detect the patient, cancer, we can characterize it, that at what stage it is, at what state is it spread, and lastly, we can monitor the progression also. In surgery, it is also we are using various AI-based models for surgery. And then it is also used in treatment, cancer treatment monitoring, that how what are the medications we are providing to the patient, how it is working, or it is effectively working or not. It is also used in drug discovery and development. Various AI-based models have been developed through which we can determine what are the various targeting sites and how, at what state they the drugs which we are working on it can treat the disease. It is also used effectively in chemotherapy. As we all know, chemotherapy has various side effects. AI-based models can reduce that side effects and can reduce the drug-dependent toxicity also. It is also effectively used in immunotherapy as well, where the AI-based models which are used are human leukocytes, antigen models, mass spectrometry database models for treating cancer. Thank you. Hello.
Yes, ma'am, you start.
Yeah, Aditi ma'am. May I? Okay, okay. Yeah, so this is Sadaf.
Yes, ma'am.
Right. Sadaf, you presented it, I mean, the presentation was quite okay, but the point is that when you say that, there are algorithms that will be designed for treatment of cancer in different aspects, right? Whether it is immunotherapy, radiotherapy, chemotherapy, whatever it is. So I was wondering, in cancer, it is mostly personalized way of medication, right? The therapy is personalized. So will your algorithm be able to serve in person?
And actually, if we talk about cancer, it is generally divided into 4 stages. So while developing these models, we can predict the changes in genes. We can predict the changes in genes by using these models, and we can predict that not right now, but in future, if is it possible that a patient can develop cancer. And if we talk about targeting models, then we can also predict that whether the drug which we are giving to the patient, will it target to that site or will it provide the treatment? Because in cancer, it generally causes the formulations which are now available in the market, they are mostly site specific. Because in cancer, the drugs which are previously there, they cause non-specific drug distribution, like the drug distributed throughout the body, that will cause various side effects. So to reduce that, we are developing these models, so which we can we can predict actually whether the drug which will go to that site only, or it will be effective to that site only or not.
Yeah, but I was wondering, my question was more directed towards, difference from patient to patient, right? But yeah,
It will
that's
be not
okay.
patient specific. It will be not patient specific, but yes, it will somehow it will help in treating cancer.
Yeah, but then how? Then the question remains the same. But it's okay. Okay, please. Aditi ma'am, you may please continue. Yeah.
So Sadaf, I have only just one concern in after listening to your presentation, was that you have given a very generalized picture of the aspect, how AI is being used. It's a very general that it can be used in these fields, that field, but it would have been very good if you would have supplemented it with some actual examples or some studies where it has been actually implemented, so it would have registered to us in a better manner probably. So I don't know if it is okay to ask or not, but can you give us any small example or anything you just to supplement what you have told us, any real life example for that?
Yes, ma'am. There is one publication in which ANOVA and random forest support models are used in chemotherapeutic treatments. There are research papers which are available.
So these are to study synergistic effect, or actually they have been applied in cancer therapeutics or anywhere. And
they are applied, ma'am.
Given the reference here also, if it is a published paper.
Ma'am,
right?
they are applied on some set of patients, and it has been found that they are effective in cancer chemotherapy. It is not available in the market,
Yeah,
but
so
it is
are
there
there
in
some
research.
genes, the molecular markers that have been targeted? Can you just, or in a certain type of cancer? Because when you say cancer, it is
Ma'am, it is, yes, ma'am, it is effectively used in breast cancer also, lung cancer also, and in cervical cancer also. There are research papers which are available.
Cancer is a very multifarious thing. It's too wide to just say cancer, It can be various forms. Anyway, it's fine. Okay.
Yeah.
Manish sir, if you have any question, you can go.
Sadaf, I just,
yes, sir.
I listen you, and then I got one question here. So,
Manish sir, you are not very clear. Manish sir, you're not very clear.
Okay, I'm close to mic. Can you hear Sadaf?
Yes, sir.
Yeah, Sadaf, I'm asking, like, if you are just going for chemotherapy like in a traditional way, and if you are using AI chemotherapy, right? So what was the, what is the problem with the traditional way that you can overcome through the AI?
Sir, in traditional way, while you give chemotherapeutic
Traditional means in
drugs,
conventional way, yeah.
yes, sir. So if we give chemotherapeutic drugs, then it will produce side effect. Like since we are not giving the formulation to the like, for example, if we are having breast cancer, then if we have to treat the only like at the cancer site only, we have to provide the treatment. But while we are giving chemotherapeutic drugs through IV route, it will circulate in the whole body, and it will create problem or toxicity to that part also, which is not having cancer. So while if we use AI models, we can predict that at what cancers, at what site or at what gene we are, or what site we have to focus, that the non-specific drug distribution will be avoided.
Okay, means targeted therapy can be done, right?
Yes, ma'am.
But how AI is being used there? You can use any, like most of the nowadays we have some drug delivery system that can even deliver the drug to the particular sites.
Sir, but
then how the AI is being utilized here, or you are using AI to overcome this targeted therapy, if you are saying, like this example?
Sir, we can, like the patients which are like treating with traditional chemotherapy, we can use their database that how the drugs are like circulating in the other parts of the body to those patients, and through that we can set a database and then we use AI there.
Okay, Sadam. Okay. Okay, good luck.
Thank you, sir.
Okay, that's all.
Hello everyone. Sorry to interrupt you all. I request to all the participants, please be limit in your time, because we are having more lectures, we are having more presentations, so please be on time limit. Thank you.
Next up, I would request Ms. Saloni Anand from Birla Institute of Technology to proceed with the presentation.
We can see the presentation. I think it can be started.
Yes, Saloni, you may go ahead. Not getting voice.
Saloni, we can see your presentation. Just make sure that the mic is not mute, right? You're not audible.
You're not audible.
Yes, you're audible.
Saloni, I think I cannot hear you, Saloni, but I don't know if other can hear.
Even I cannot hear her.
No, even I cannot hear Saloni's voice. Ananya, can you hear? We can't hear the presentation. Yes. We can see the presentation.
Yes, Saloni Anand. Ma'am, are you there?
Yes, Saloni.
Saloni, please continue.
Miss Saloni?
Saloni, open your video first. Only then we'll be able to communicate with you, I guess.
Seems some network issue, bhai.
Maybe she cannot hear us.
Ma'am, we are sorry, but we cannot hear you.
Sonali, your video is on, but we're not able to hear you. Are you trying to speak something?
Your slides are changing, but
Can we go to the next one till she's fixing her issue?
Sure, ma'am. Miss Saloni, you can unshare the screen. We'll get back to you later. Next up, I would like to invite Miss Palak Sachdev for a presentation.
Palak, we can see your presentation, can't hear you.
Again, I think you cannot hear.
Am I audible?
Yes, yes, Palak. Please go ahead with your presentation. Palak.
Am I audible?
Yes, Palak.
You're audible. Why don't you start? Start presentation.
A very good afternoon. A very good afternoon to our esteemed judges and all of you. My name is myself Palak Sachdeva from BSc Honors Microbiology, 3rd semester, Amity Institute of Biotechnology. The topic of my presentation is AI-driven advancements in fermentation technology. So, let's start. The incorporation of artificial intelligence in fermentation research. Just a second. Yeah, the incorporation of artificial intelligence in fermentation research is transforming the business, providing several benefits while also posing new problems. Artificial intelligence is increasingly being utilized to regulate and to monitor and regulate the fermentation process, resulting in greater efficiency and uniformity. For example, artificial intelligence can forecast the optimal fermentation temperature, pressure, and nutrient levels, lowering the chance of human mistake and enhancing overall production. Despite several advantages, incorporating artificial intelligence into fermentation research also has some problems. One of the most significant barriers is the high expense of integrating AI technology. AI system creation and maintenance need major financial investment, which may be prohibitively expensive for small firms, and the complexity of AI systems also needs a high level of knowledge to successfully operate and manage them. Another possibility is that AI replacing human workers in the fermentation business. However, while AI can execute some activities more effectively than humans, but it cannot replace the creativity, intuition, etc. Despite these obstacles, the benefits of integrating AI into fermentation science are apparent. The use of artificial intelligence has the potential to significantly increase the efficiency and quality of the fermentation process, thereby revolutionizing the industry. Next, what are some of the advancements and applications of using AI in the fermentation process? Bioprocess optimization. Large data sets can be analyzed by AI algorithms to determine the best circumstances for microbial growth and product creation. This results in increased yields, lower production costs, and shorter fermentation times. Strain development, AI can forecast the effect of genetic changes on microbial strains. This speeds up the development of customized strains with desired characteristics like increased product yields, improved tolerance to unfavorable environments, and improved product quality. Real-time monitoring and control, artificial intelligence-powered sensors and monitoring systems provide real-time data on fermentation factors such as pH, temperature, and oxygen levels. This allows for instant modifications to ensure optimal conditions and the detection of deviations. Next, predictive modeling. Based on numerous input factors, artificial intelligence models may forecast the predicted yield of fermentation process. This helps with resource planning, forecasting production, and decision making. Then scale up and downscaling. Next is genetic engineering and synthetic biology. AI can help with the creation and optimization of genetic constructs for fermentation microbes. It aids in the development of bespoke strains that efficiently produce desired goods. Next is accelerated research and development. AI-powered technologies can dramatically accelerate fermentation process research and development, allowing scientists to investigate a wider range of conditions and parameters more quickly. Drug discovery in biopharmaceuticals. AI can help in the discovery and synthesis of biopharmaceuticals via fermentation, hence speeding up the development of new therapeutics and vaccine. Overall, artificial intelligence-driven breakthroughs in the fermentation technology are increasing efficiency, consistency, and innovation across a variety of industries, with the potential to produce new products while reducing the environmental footprint of traditional fermentation processes. That's all. Thank you.
So again, Palak, you have just given an overall gist of the aspect, whereas a very interesting topic you have picked up, that how AI can be integrated in fermentation, but it would have been very good if you would have integrated some actual examples where it has been used anywhere, or is it just in books or in labs or in research phases, right? So can you think of any example where it has been integrated, AI has been integrated?
Yes, ma'am. There's an AI software named Fermento that uses different concepts to improve the fermentation process by providing proper methods and using and giving us an idea of how to do the process so we get the maximum yield.
Okay, so one more thing you have just said initially, you have told few negative points, like it can be very financially expensive and all. So
Yes,
what do you think, in your opinion, that whether AI should be integrated or not?
Yes, ma'am, it should be integrated.
Even if it is expensive?
Ma'am, everything has certain advantages and disadvantages also, but the advantages are on a wider side, so it should be used.
That's all from my side.
Okay. Yeah, so Palak, in addition to what you just said, I mean, I was going through your presentation, and one of your points you mentioned that, indirectly it mentioned that the human labor can also be reduced by utilizing AI,
yes,
right? Fermentation process. So now I have two questions in my mind. One, when already so much sophisticated automatic, machines or instruments are already there to, undergo the process, then why would you require AI? And if in a country like India, you are utilizing AI, which is also, like Aditi ma'am also pointed out, though you answered it very smartly, that okay, even if it is cost effective, it doesn't make difference once it is more, beneficial, right? So, wouldn't it affect where people are so much, in a country like India, where we really need so much of employment for our younger generation, and putting up AI on top of it, which will, take up all the, the factors that can be governed by themselves. So how good it is and how relevant?
Ma'am, it is AI is useful because it gives us maximum yield. It gives us the methods so that we can increase the production, better quality. So therefore, it is useful in that sense.
Quality in no way is compromised now also, right? Even now when we go with, different automated machines, quality is not compromised here. So
yes, ma'am.
But that's okay. Yeah, Dr. Manish, you please continue.
Okay. I heard your presentation. So, the fermentation technology or fermentation process in conventional way, right? So, in AI, if you're putting like AI or if you're using AI here, so what are the major steps that you control or that you can basically regulate using AI in fermentation? Fermentation is not a simple stage process; it has a different stages. Like it's how it is going to control each and every step, like in fermentation, and then overall it's going to increase the yield.
So we can monitor and regulate temperature, optimal temperature, pH and pressure conditions that are required for fermentation.
Okay.
And
how AI is going to do if you are like, if you are going to do like with the manually or using some systems or some operators and all, and how you are going to put AI here, how you are going to use AI. What are the basic needs here you will basically need in order to use the AI for the fermentation? What will be
please
your requirement here?
So please repeat, you are not audible.
What will be your requirement here in order to use the AI for the fermentation? Is how you make the certain setup for the AI. Because you are just saying like AI can regulate this, AI can do this, AI can that. So do you think like that what are the major requirements or basic requirement here in order to develop certain models for the AI?
So basic requirements.
Yeah.
So
you can do the temperature control, you can control the humidity, you can do many other things.
pH and all. That is.
Yeah. But what are the what you know about like what you would need in the first stage in order to develop a model for it? Any idea?
No, sir.
Okay, I think you should like go through it, and you must have for many articles. So try to understand like what are the basic requirements for that.
Okay, sir.
Okay. Okay, Palak. Good luck.
Thank you, sir.
Hello. I would invite Ms. Namrata Kumari from Birla Institute of Technologies to proceed with the presentation.
Hello, am I audible?
Yes, you are.
Okay, I'm sharing my screen. So is it visible? Hello.
It is visible. Put it on a presentation mode and start.
Okay. So good afternoon, everyone. This is Namrata from BIT Meshra, a Masters of Pharmacology student. And today I'm going to present on the topic analysis of proteomic changes in the rat plasma in response to hypobaric hypoxia. This will be the overview which I will be discussing. So moving to the important part, what is hypobaric hypoxia? So whenever we move to the high altitude, because of the high altitude, there is decrease in the environmental pressure, or we can say that there is a decrease in the barometric pressure, due to which there is low inspiration and the people face the condition of hypoxia called as hypobaric hypoxia, or sometimes it is also called as high altitude hypoxia. In case of mild to moderate hypobaric hypoxia, it is characterized by hypoventilation, fluid retention, and the sympathetic drive. And in case of moderate to severe hypobaric hypoxia, it is associated with the edema in the brain and the lungs, and sometimes causes the death of the people. So the treatments which are available for the hypobaric hypoxia includes symptomatic treatments like analgesics for headache, promethazines for the nausea and vomitings, and the glucocorticoids like dexamethasone. And for the acclimatization, a popular group of drug which is being used are carbonic anhydrase inhibitors like acetazolamide and methazolamide. Now, these are some of the complications which are associated with the hypobaric hypoxia. Hypobaric hypoxia may lead to tachycardia.
Slide is not moving.
Yeah, your slides are not moving.
Okay.
Just shift your slides 1-2 times up and down. Yes, moving now.
Is it visible now?
Yeah, you are on the 4th slide, right?
Complications.
Yes.
Okay, so moving to the complications which are associated with the hypobaric hypoxia, it includes tachycardia, pulmonary edema, which is also called as high altitude pulmonary edema, obstructive sleep apnea. In the case of hypobaric hypoxia, there is a case of which leads to the sleep apnea, which is also known as obstructive sleep apnea, cerebral edema, and there is a sometimes formation of the reactive oxygen species in our brain, which can further lead to the precipitation of different neurological disorder. Due to the increase in the cellular oxidative stress, which will lead to the damage in the lipid, protein, and the DNA of our body, and may further affect the cellular functions of our body. Hypobaric hypoxia are complex and characterized by the alteration in expression of number of the genes and the protein in our body in order to maintain the homeostasis of our body. Now there are different evolution parameters. It is moving. My slides are moving. Hello?
It did not move after complications. Yes, now it is moving. Evaluation parameter.
So moving to the evaluation parameters to check whether there is a condition of hypobaric hypoxia in animal model, there are different parameters which can be checked. First of all, moving to the behavioral studies, which includes the plus maze apparatus, object recognition test. Second, moving to the physiological test, which includes respiratory rate, checking of heart rate, blood pressure, hematocrit level, etc. A biochemical assay includes inflammation test and level of the different cytokine in our body, and different histopathological studies of the organs. So why there is a need of analysis of proteomic changes in the case of hypobaric hypoxia? So to study the proteomic changes in the rat plasma, it will contribute to the understanding of adaptive mechanism of the living organisms under the low oxygen condition. It will help us to know about the alteration of different genes in the case of hypobaric hypoxia and the potential biomarkers which are associated with the hypobaric hypoxia. It will also help us to provide the insight of the molecular pathway that are involved in the body response to the hypoxia, and it will act as the instrument which can help in the development of different therapeutic strategies for prevention and the identification and as well as the management of different hypoxia related disorders. So there are different methodologies which can help us in the analysis of proteomics. First, I have enlisted a different methodology. First will be the exposure of hyperbaric hypoxia. Here the animals which we are taking, for example, rats, are first introduced or experience the hyperbaric hypoxia condition in the hypobaric chamber at the stimulated temperature of approx 7.
To kindly conclude the presentation as your time for the presentation is up.
Second is sample collection, and after that there will be the plasma separation. After that protein extraction will be there, and protein quantification. Therefore, after that there will be the separation of the protein. For analysis, there will be the mass spectroscopy analysis, and after that there will be the validation process using the established method like ELISA and all. So moving to the hypothesis or conclusion part, after conducting different bioinformatics studies, which for the hypobaric hypoxia, it will act as a unique biomarker in the hypobaric hypoxia settings and may offer the fresh prospectus for the management and for the treatment of hypobaric hypoxia. Thank you.
You have not shown any results.
Ma'am, actually, I'm doing this experiment in my college, and so I have not put any result because I have just started working on it.
You have just shown the shown your hypothesis here. You have just presented your hypothesis.
Yes, ma'am, because I'm working on this, and I have only done some of the literature searches which I have mentioned here.
Okay, Gurjeet ma'am.
Yeah. In fact, Namrata,
yes,
so
ma'am.
your thought process is what you have, shown us, hai na?
Yes,
but
ma'am.
the point is, like in one of the slides you said, I mean, I'm more on technique aspect. So my question to you is, you said you're going to precipitate the protein with TCA.
Yes,
right?
ma'am.
10% TCA is what you are going to use.
Yes,
and
ma'am.
then you are suggesting a technique that is two-dimensional.
2D electrophoresis. Yes, ma'am.
Will you be able to get any result when you are doing it precipitating with TCA? You will get two-dimensional results. What are
Ma'am, I have separately mentioned about that we will be doing the 2D electrophoresis for the fractionating or for the fractionation of the DNA, for separating them for protein.
Fractionation.
For separation of protein.
But which method of fractionation are you going to use?
Ma'am, 2D electrophoresis.
Okay, thank you, Namrata. I think
that you
you
discussed
really
it,
need
na?
to
Now
work
you
on
will-
the technical aspect.
you will have an
if
insight.
you want to execute.
Again,
Namrata, can you fractionate the protein using 2D electrophoresis?
Yeah, that's what that is.
You know what is the purpose of doing the 2D electrophoresis? What you can do with the 2D and how you perform it.
Sir, as far I know, for 2D electro 1D and 2D for 1D, we used to separate the DNA molecules, and 2D is based on isoelectric
You are doing 2D, right? If you are doing 2D, it must be first you have to do the gradient, electrophoresis, and then you will go for this another one or a vertical one.
Yes, sir.
Right. So, and in that case, you will see the the expression level of that particular protein, like after even 2D, and then you will get a certain gel pictures, and then you can find
You
out.
really need to work a lot on it.
Okay.
How you're
Okay,
going to
ma'am.
execute
Sorry,
the
I was not having so much idea.
It's okay.
Okay, Namrata, that's. I think you must read it before you are going to present the data, right?
Okay, sir.
Yeah.
Good
Okay,
that you
Namrata.
presented it here. Na? You presented it
Yes.
here, and you got a lot of insight to look
Yes,
into
ma'am.
your methodology again, isn't it?
Yes, ma'am. Thank you. Thank you, ma'am.
Okay, Namrata. Best wishes.
Okay, Namrata.
Best wishes.
Next, I would invite Priyal to proceed with the presentation.
Good afternoon, everyone. I'm Priyal from BTech CSC, presenting my topic on.
Go to the first slide and just show the title.
Slide.
Artificial intelligence for ambient air quality analysis. The possibility that artificial intelligence has to change ambient air quality regulation is examined in this research. We provide a comprehensive analysis of accepting AI application, highlighting the advantages and disadvantages server models of air quality prediction and control. These encompass several AI approaches such as machine learning and deep learning. The usefulness and efficiency of AI applications are evaluated by analyzing real-world case studies. AI has a lot of potential, but there are obstacles to overcome when putting it in practice, such as scalability, interpretability, and data needs. We examine the potential applications of AI in air quality control in the future, analyze these problems, and offer potential remedies. The goal is to offer a thorough grasp of the part AI may play in environmental management, as well as route map for the rising is use. This report calls for more investigation into the potential of artificial intelligence to develop long-lasting and efficient air quality management system. Now, I would like to introduce this. Efficient air quality control crucial for public health, the environmental and economic stability. It harms climate change. AI, particularly machine learning and deep learning, is increasing use in environmental sciences. AI analyze vast data from sources like satellite, monitoring stations, and meteorology data to predict air quality and identify complex patterns. It contrasts to conventional rule-based algorithm. Machine learning algorithm allow computer to learn from data inputs and produce results that fall within a given range, while statically analysis to generate truthworthy and repeatedly judgment and outcomes. They basically learn from previous calculations. Here we have some examining air quality using AI applications. Extensive study has been conducted to further examine that effectiveness of environmental marketing as a tool to support AI involvement in ambient air quality controls. The result has been positive and the potential for real-world application has advanced. Various AI techniques has been applied to improve air quality management, monitoring, and prediction. Like in one noteworthy work, a deep learning algorithm called long short-term memory to predict Medellin, Colombia air pollution. The study predict hourly PM 2.5 values using methodology and historical air quality data. High accuracy was demonstrated in the data, and the model outperformed convenient time series forecasting techniques by sustainable margin. LSTM are predominantly used to learn and classify sequentially data because these networks can learn long-term dependency between time steps of data. It includes video analysis, sentiment analysis, etc. In a different research, estimate ground level PM 2.5 concentration throughout China using machine learning technique and satellite data. Third one is forecasting the air quality index in California, USA across the Pacific California. I state regularly affected by wildfires that affect air quality used artificial intelligence to predict the air quality index. Now we have CNN is an example of deep learning system that excel at processing picture utilizing the CNN model. The research examined satellite imagery to identify characteristics associated with air quality such as smoke plumes and wildfires. Basically, CNN is a step-way algorithm. We can divide this algorithm in 6 steps. Firstly, we load an image and resize it to a predefined image such as 224 into 224 pixels. Scale the value of that pixel, and the 4th steps is to select a pre-trained model, run the pre-trained model, and display the result. Here we have some advantages of AI in air quality control. First one is increased
You're kindly requested to wrap up the presentation.
The advantages are like real-time monitoring, predict analytic, early warming systems, In real-time monitoring, AI system can continuously monitor air quality and provide real-time data, allowing for rapid detection and response to air pollution, event or change in quality. In data fusion, AI may combine information for several sources, including historical records, remote sensing, and ground-based sensors. Now we have disadvantages of AI. Algorithm complexity. AI models can be complicated and challenging to understand, particular deep learning algorithm. It is difficult to comprehend how this model operate on the inside, which make it difficult to define and explain the prediction they make. Energy consumptions. Deep learning the model training may be an energy intensive process, which may have an impact on the environmental, and AI infrastructure is not run on renewable energy sources. Now let's move to the conclusion. In conclusion, artificial intelligence has the potential to revolutionize efficient air quality analyzed by enabling real-time monitoring, predictive capabilities, and data-driven decision-making. However, it comes with challenges such as cost, data quality, and ethical consideration that need to be carefully addressed for this effective and reasonable implementations. That's all. Thank you.
Okay, I think we are really running out of time and we really need to wind up. So I think either of us can ask question. Ma'am, please, yourself.
I thought you are about to ask. I was also about to say this thing that we'll just stick to one question per presentation. So any one of us can ask question. Manish sir, if you wish to ask anything.
Just one question here. Like here, I don't remember the name. Priya. Yes, Priya. Here air quality means what you are going to include in the air quality. What parameters? What kind of things you are going to include here? In you meaning like what is the meaning of air quality here for you? Like air pollutions or gases or whatever, or any particular metals or any particular
matters, particulate matter. These are tiny particles in the air that can be inhaled into the lungs that have adverse health effect. So basically, we have to check that through our AI that it is present or not, or ground level ozone, that the earth surface can lead to respiratory problems and other health issues. Nitrogen dioxide, a gas that can irritate the lungs and lower resistance to respiratory infections.
Okay, got it.
Oh, thank you,
Okay.
sir.
Okay. Good luck.
Next, I would like to invite Muskan Gupta from BITS to continue with the presentation.
Hello, am I audible?
Yes, Muskan, you're audible.
Okay, I'll be sharing my screen. Is it visible?
Yes.
Good afternoon, everyone. Good afternoon, everyone. Today, I'm Muskan Gupta from the IIT Meshra Ranchi. Is going to present on radiant solutions eliminating the path of obesity with photodynamic therapy and selective obesity. The topic of today's presentation is my current master's research work. So here I will be discussing about obesity and how I have linked it with photodynamic therapy. Starting with what is obesity? Obesity generally occurs due to disruption between the energy intake and energy expenditure. WHO defines obesity as abnormal or excessive fat accumulation that presents the risk to health. Here is a graphical representation of rank of different countries according to its obese population. India ranked third among it with 44 crore people obese. Now, generally, people misinterpret that obesity only affects physical appearance, but Overweight and obesity are the fifth leading risk of global deaths. The risk factor of occurrence of several diseases like respiratory, reproductive, pancreatic, etc. increases when the person becomes obese. Next, there are various parameters to evaluate obesity. First is BMI, body mass index. It is a numerical measurement which categorizes individual according to its body weight. DEXA, dual energy X-ray absorptiometry. Here, different intensities of X-rays are used to produce medical imaging of bone, lean tissues, fat tissues, etc. Next is waist to hip ratio and waist circumference. These are anthropometric measurement of to assess body fat distribution. Next is bioelectrical impedance. It is a method to measure the resistance of the body tissues. What happens here is when a person becomes obese, then the lipid droplet size increases. So when the current is passed to the body, the more it faces more resistance because lipid droplets are more. Now, there are medical interventions to treat obesity. First is pharmacotherapy. FDA has approved 5 drugs, but it comes with certain limitations like weight gain after discontinuation, gastrointestinal side effects and malignancy, etc. On the other hand, surgical intervention includes bariatric surgery, which is recommended for patients which have BMI more than 40. Now, this also comes with certain limitations like potential long-term nutritional deficiencies, dumping syndrome, etc. Now, as we have discussed, these both interventions come with certain limitations. So we need something which is more selective to this treatment. One such treatment which I have discovered while doing literature review is photodynamic therapy. Photodynamic therapy is a modern, minimally invasive form of therapy, which is based on local or systemic application of photosensitive compound or the drug, which is intensely selectively accumulated to the targeted tissue. Here, what happens is, first drug is administered, then light of specific wavelength is irradiated so that the drug gets activated. Then it undergoes photodynamic reaction with the dissolved oxygen present inside the biological tissues and forms and causes cell death. Now, what happens is first the photosensitizer is in the ground state. After light irradiation, it goes to singlet excited state. Then it goes to the triplet excited state through intersystem crossing. Here, what happens is the spin of the electron of photosensitizer is same as that of the ground state. So it is unable to come back to the ground state. Then being highly unstable, it undergoes reaction. Basically, 2 types of reaction occurs, but mostly type 2 reaction occurs, which forms singlet oxygen and causes cell death by apoptosis due to oxidative stress. Now, I have, according to literature review, I have decided to work on aminolevulinic acid. Aminolevulinic acid is an endogenous amino acid which is synthesized during heme biosynthesis pathway. Now, 5-aminolevulinic acid is metabolized to protoporphyrin 9 in mitochondria, and it is a photoactive compound that is photosensitizer. Another thing that which has been found that it is 3T3L1 selective, that is adipocyte selective, so that it will selectively get accumulated inside the adipose tissue. It has been found that it gets activated when irradiated with a wavelength of 650 nanometer. Now, if we see the spectrum of light, 650 nanometer comes inside the red light, and if we see the penetration of the light to skin surface, then red light can easily penetrate up to subcutaneous region where fatty tissues are present. Moving forward, the rationale is that due to above limitations, both pharmacotherapy and bariatric surgery, photodynamic therapy can be used. And according to literature's review, it has been found that ALA has anti-obesity property, which has been neither explored nor established through PDT in obesity. So my hypothesis is that aminolevulinic acid gets accumulated. If aminolevulinic acid gets accumulated, and after irradiation,
Please wind up the presentation as your time for the presentation is over.
If aminolevulinic acid gets accumulated and after irradiation, if it gets activated successfully and releases reactive oxygen species, which leads to apoptosis of excess adipocytes, then it may be a choice in future for treatment of obesity, as it is minimally invasive procedure and has least to no side effects. This is my methodology. First, analytical elucidation of ALA will be done. Then I'll induce obesity in urine model to develop hyperplastic obesity. Then I'll be exploring anti-obesity property of ALA PDT, and I'll evaluate ALA PDT on the basis of different physical, biochemical, and histopathological parameters. These are my references. Thank you.
Muskan?
Yes, ma'am.
Okay. So yours is also just hypothesis you are working, you have not started working on it?
No, ma'am, I have not started working on it. It is just I have just 3 months have passed, and currently I am at ALA elucidation, and I have started the development of obese model.
So for making your methodology, you must have undergone literature.
Yes,
so
ma'am.
is the similar literature already available where people have worked for obesity, this photodynamic method for obesity?
Ma'am, actually in 2023, June 2023, a single patent has been filed for fluorine E6 mediated photodynamic therapy. No work has been done for aminolevulinic acid based photodynamic therapy, and I am targeting on hyperplastic obesity. The work which was earlier done is on hypercellular obesity.
So what will be the advantage of doing this? If you like, if you will do this work, how it will add up to the existing knowledge?
Ma'am, first of all, this PDT is a selective treatment. Like it will only target to the adipose tissue because it will get accumulate only in the adipocytes. So there will be least chances of other side effects. No other organs and will be involved and will not play a huge role. And it will be a minimally invasive method. No other side effects can be there. As I have discussed earlier, that there are limitations of both pharmacotherapy and bariatric surgery. So people who are unable to reduce their weight, they can, if it is successful, then PDT can become a treatment option for obesity.
So, Manish sir.
Okay, from my side, I
Okay. Okay, thank you, Muskan. All the best.
Thank you.
Next up, can we please have Ms. Saloni Anand from BITS?
Yes, ma'am. Am I audible?
Yes, you're audible. Saloni,
Okay.
please constrain yourself to the time limit.
Okay. Is my screen visible?
Yes, go ahead.
Good afternoon, everyone. Myself, Saloni from BIT Mishra, Ranchi, and my today's topic of oral presentation is gene expression analysis in diabetes. So, coming on the introduction part, what is diabetes? Diabetes refers to a group of disorder
Can you please
characterized.
skip the introduction? You can
Okay,
skip
ma'am.
the introduction, right? Yeah, you can skip all this. Go to your objective and yeah.
So for the gene expression in diabetes, we will be following several key steps. First, sample collection. For this, we'll obtain tissue or blood samples for both diabetic and healthy animal model for analysis. Then RNA extraction, we will isolate the RNA from the collected sample. Then for RNA quantification and quality control, we will assess the quantity and quality of the extracted RNA using techniques like spectrophotometry and gel electrophoresis. Then we will do for the cDNA synthesis, that is the reverse transcription. In this, we will convert the extracted RNA into complementary DNA using reverse transcription. Then we will do the gene expression profiling using techniques like PCR, microarray, or RNA sequencing. Then we will do the data analysis. In this, we will analyze the gene expression data using bioinformatics tools and software. For pathway analysis, we will investigate the biological pathway and network associated with differentially expressed genes. Then we will do validation. We will validate the results obtained from gene expression profiling using independent techniques. And at last, we will integrate with clinical data with clinical information such as patients' demographics, disease progression, and treatment history. So my rationale of this choosing this topic is, in this we will compare the gene expression between the healthy animal model and the diabetic animal model. There are certain susceptible genes which are responsible for the diabetes, and these are the following: TCS7L2, PPARG, KCN, Q1, CAP, and 10, which was the first diabetic genes to be identified through a genome scan. Then hello.
Is that all?
No, ma'am. I'm not able to. Ma'am, wait. Then IRSI, ABCC8, HNF1A, HNF1B. So the hypothesis of my project will be based on the above plan of work for the research work. We will get an idea regarding that which particular gene is responsible for causing diabetes. And also this particular gene will be further analyzed, that whether this gene is overexpressed or whether this gene is suppressed, and by targeting it in my future research work. Thank you.
So again, you are just suggesting that all these genes are going to give you some, outcome, but we do not know whether you will be getting any result out of it or not. And another of my concern is that diabetes already has been explored a lot, and it has been studied since, now decades. And how and what, why you have chosen these genes? क्या इनमें कुछ काम हुआ नहीं है अभी तक? अगर हुआ है तो किस लेवल तक? And what was your aim of taking these genes particularly?
Ma'am, these are the genes only which are responsible for causing diabetes. So we will study on them individually. I have not yet started my project, but I have just made a hypothesis that these are the particular genes that are responsible for the diabetes. And when we'll compare the genes between both, then we will get to know that which is the particular gene that is responsible for causing diabetes.
Manisha?
Yes, Saloni, how many genes you are going to consider for this work?
Sir, I have mentioned some of them, but there is not a clear idea how many genes I will consider, but it will be in between these only.
Okay, okay.
Okay, sir.
Okay, Saloni. Okay, good luck.
Thank you, sir.
Okay, we can move to the next one.
Next up, can I please request Ms. Ayushi to present? Ms.
Yes.
Ayushi.
Am I audible?
Yes, you are.
Okay, let me share my screen. From where to share the screen?
There is an option share screen.
Where? Yes, I found it. Is my screen visible?
Yes, it is visible.
Okay. Okay. So a very good afternoon to one and all present in this online international conference. My name is Aishi. I'm a research scholar of biotechnology. And today in this event, I would like to present before you all one of my proposed innovative idea, which is a human body studying robotic, magnetic, and X-ray system. So first of all, I would like to talk about that particular existing technology, which kind of inspired me for thinking about this innovative idea. So that existing technology is nothing but computed tomography scan, which is also known as CT scan. As we can see the CT scan machine in this on the screen. So I would like to talk about what is a CT scan. It is a basically imaging technique which makes use of the radiological study, which uses the radiological study. Radiological study means it makes use of certain radiations to form the images of the inner body parts. So the main radiation which it uses for the imaging of the inner body parts are the X-rays. So basically how it works, if I talk about in a very brief, then basically as we can see in this figure, in this circular part there are multiple X-ray tubes attached. From those multiple X-ray tubes, multiple X-rays come out, and as this circular part rotates, the X-ray coming out of the X-ray tube also rotates around the patient's body, and that's how the image of the complete human body can be made using this technology.
So basically, it makes use of multiple X-rays rotating around the human body to form the clear images of the inner body part. And it is a very effective technology for diagnosing the complete body whenever required. But it has also has certain drawbacks associated with it. One of which is it is very expensive. This medical diagnosis is very expensive and not affordable by every patient, and it can also greatly harm human body, because so many, when we get our X-ray done, the amount of X-ray radiations which enter into our body at that time, 300 times more X-rays enter into our body when we get our CT scan done. So it can be very harmful on the human body, and we must get it done only if doctors suggest us. So basically, my motive behind this proposed innovative idea is to bring CT scan, to make CT scan portable and bring CT scan in every doctor's chamber, so that the diagnosis of complete body gets relatively easier as compared to now. So what is my proposed innovative idea? Human body studying robotic, magnetic, and X-ray system. So basically, the proposed idea of mine will contain two parts. The proposed innovative idea of mine will contain two parts: an AI-based robotic objects and an iron ring in which there will be embedded multiple X-ray tubes. Let me show you. So this is my innovative idea. Here in this innovative idea, here we can see there is a robotic object or a robot, and this is the iron ring, and these blue colored things are nothing but the multiple X-ray tubes from which X-ray are coming out, and this is the human body. So basically, how this machine will work, if I talk about how this machine will work, basically the iron ring containing multiple X-ray tubes will surround the human body, and the X-ray coming out of the X-ray tubes will be exposed towards the human body. And how will this iron ring rotate? This iron ring will rotate. The robotic object in its hands will have strong magnets. With the help of that magnet, the robotic object, without touching the iron ring, will rotate the iron ring. And basically, what will happen? The iron ring will
presentation has only 30 seconds are left.
The iron ring will contain the facility for producing high voltage, so that cathode ray can be emitted in the X-ray tubes, and by the cathode ray, X-ray can be produced and enter into the human body. So the robotic object will not only be able to rotate the iron ring, but it will also be able to track the X-rays which is entering into the human body. It will be able to visualize the human body parts using that X-ray, and other than that, the robotic object will also be able to diagnose, make the diagnosis and analysis of the abnormalities or diseases in the human body. So that's how the artificial intelligence will help in my proposed innovative idea. Now this is how the X-ray tube this will be the instrumental setup of X-ray tube from inside. There will be small X-ray tubes, not large X-ray tubes, and this will be the setup in the X-ray tubes. And this is my innovative idea. Here the robotic object is emitting the magnetic.
Time is up. Kindly move to the conclusion now.
All right. So this is my innovative idea. Now, if I talk about what would be the merits of my proposed innovative idea, then these will be the merits. That is, there are many diseases such as cancer, which is diagnosed at a very advanced stage, at a very later stage, when it becomes quite impossible to treat it. But if such a technology will be available in every doctor's chamber, every physician's chamber, then maybe this problem will be solved, and there will be lesser cases when the diseases are diagnosed quite late, and maybe diseases will be diagnosed at early stage, and we will be able to treat those diseases. And since this technology will be cost effective also as compared to actual conventional CT scan, and since this technology will be smaller than CT scan, so it will be probably less harmful than the on the human body as compared to conventional CT scan. So these are the merits or the advantages of my proposed idea. So that was all. Thank you so much.
Okay, Ayushi, I congratulate you for your idea. But as far as I know, there is already a portable CT scan available in the market. And I would just like to know, what do you mean by this portable CT scan? Is it going to the patient bed, or is it going out of the diagnostic building to a hospital, another area of the hospital? What do you mean by that?
Ma'am, by the word portable, I actually mean that it will be available in every doctor's chamber, and whenever the doctor would think that complete diagnosis of patient is required, the doctor will use this particular technology for the diagnosis in its chamber itself.
So you think each and every doctor will be able to do the CT scan, or a radiologist would be required to do the CT scan? As far as I know, these imaging and all these are also done by specialists who are called as radiologists, and not each and every doctor are authorized to use the CT scan, first thing. And the other is that X-rays are supposed to be harmful as it is, so making it like doing it very randomly or in all and every patient, whether it is required or not, I have a doubt whether how much merit it holds for this, right? And most importantly, that a portable CT scan is already available. You must look into it, how it works and what all is there. Yeah. Yes, ma'am.
Yeah, that's true. That's true, ma'am. So I think, Ayushi, we can take up your case a little later, because we still have so many students to go. Okay. Yeah, please. Let's start with the next candidate. I'm sorry, Dr. Manish.
Okay, no problem. No problem. That's fine. Even I wanted to ask.
Yeah, please.
Okay.
Thank
Good
you,
luck,
Ayushi.
Ayushi.
Thank you, sir. Thank you.
Yeah, organizers, we can have the next candidate.
I'm really apologetic to intervene between the session. I'm really apologetic. Actually, my eminent judges, actually, we are running the short of time, and we are having near about 5 speakers more, 1 or 2 from CSIR. So I will be grateful if you just short the question hour session. So it will be my very personal request from my eminent judges, please.
It's okay, ma'am.
Thank you.
Next up, I would like to invite Mr. Jishan Arif from CSIR IITR.
Thank you. I hope the screen is visible. Let me share my screen. Yeah. I hope the screen is visible to all. Am I audible?
Yes, Ishan, please continue.
Oh, thank you. So good afternoon, everyone. So
Just
I'm
make your
Zishan.
presentation full screen mode.
Yes, sorry, ma'am.
It's okay. Yeah, please go ahead.
Okay. So good afternoon, everyone. I'm Zishan Arif. So I will be very brief in my presenting, and thank you for the opportunity to for be a part of this conference. So I did my bachelor's from MIT, so that is also I would like to know. And presently, I'm pursuing my PhD and already completed. So to be brief, I will start with like to bridging the data gap and AI's artificial intelligence role in high throughput biological research. So like artificial intelligence is everywhere nowadays, very booming, and it's a fascinating field. So it will give you a cutting edge concept that it has the potential to improve the quality of life of human beings. And it will, if we are talking about in terms of its applications, so like it is being used as a cost effective and customized medicine, gene editing, and radiography. And in every biological domain, you can find AI is everywhere. So let me go through that. What is high throughput biological research? Since we have lots and lots amount of data, but handling is kind of a very tedious task. So it involves the use of that, how these automated technologies that generates a large amounts of data within a short period of time. So this approach now applied in revolutionized in the field of biology, that will help researchers to study the complex biological networks, pathways, and to identify new targets for the drug discovery process. So we have ample amounts of data that has been generated through the biological research and technologies. So it will give you so that is a challenging task for researchers or the scientific community. So there is the use of developing and advanced computational tools and techniques to help researchers to get some insights about that makes sense that how this data can be beneficial for the human mankind and for the society. So that AI, that is artificial intelligence, has this potential to transform this field of biological research that will help researchers to analyze large sets of data. So data challenge. Now this is another task, that how the data has been overloaded with our different domains. We are getting the data from experimental, from studies, from articles, or from any research, either from a government institute or from a scientific institute. So data integration is a very important step, and it's a major challenge through requires computational resources and well expertise personnel. So now how this AI will help you in your acceleration of your biological research. So this will help you. The AI is already the machine learning approaches or the supervised and unsupervised learning that will give you and building a comprehensive predictive models for your models. So you don't have to go for a rat or rabbit experiments for sacrificing animals in your experimentation. It will require a cost-effective as well as time consuming, and it will give you a better predictive models. So that has been used in AI has been used in a variety of ways, including biological research, from drug discovery to genome editing techniques, and these machine learning algorithms that is help you to identify that, finding out some patterns on the basis of the supervised and unsupervised learning. So that will help you that how you can transform your research through the help of AI. So these are some applications like AI has been used in drug discovery, and these machine learning approaches that I already explained in my previous slides. So it will be cost effective, and this will allow the researchers to test thousands and millions of compounds in the lab without having to sacrificing for animals. It will save your time and the resources. And AI has also been used to screen millions of compounds. Yes, sir?
Your slides are not moving. Can you just shift your slide up and down? Maybe it's not moving. Or you may continue, then maybe we can.
Its screen is visible, sir?
Yeah, screen is visible.
Slides are not moving.
Yeah.
Now it's moving, sir?
Yes, I think it's moving. Yes, moving now. Go ahead.
Okay. So the virtual screening that we can screen the millions of compounds and predicts which ones is more likely to bind with the specific targets. So this will accelerate your drug discovery process. There are also ethical considerations that has been associated with these techniques. So we also always have to keep in mind whether from where we are getting our data. From experimental institutes, we have to be very ethically correct while developing our predictive models. So these issues include like data privacy, biases, algorithms, which algorithms we are going to use in our method, and whether linear regression based models, random forest regression, or any supervised or support vector regression based models. And this AI has the potential to replace human researcher, but we have to take in consideration that how this data is coming from. So it is very much important to address these concerns so that can be that AI is used in a responsible and ethical way. So to sum up and finalize, to conclude, like this AI, that artificial intelligence has a very high in high throughput biological research, has the potential to revolutionize the field and accelerate your scientific discoveries. So through automated data processing and analysis, This AI can help and assist scientists to extract some meaningful information in a short period of time and to get some amount and to handle vast amounts of data in a short span of time. And it is also very much important to, we must have to prioritize our collaboration between the computer scientists, biologists, and other experts to use or to apply these computational tools and techniques to serve the scientific community in a better way. So with there are some with continued investment and some innovation techniques, this is some the path to transform your biological research and lead to new breathtaking discoveries and to treat some human diseases, and that will serve as a better prospects for the benefit of society and the mankind. So thank you very much.
Manish sir, would you like to ask any question?
No, ma'am. I think I had his presentation.
Okay, Gurjeet ma'am, because it's for me, it's a overall, application of AI in biological data, or you can say the handling of data with the help of various tools and how it can help in this. It's a broad and broad topic. I don't find much to ask. Gurjeet ma'am, if you wish to ask anything, or we can move to next one.
I think we can move next one.
I think she's busy. Okay, Zishan, thank you so much.
Thank you, ma'am. Thank you.
Okay, thank you. Good luck, Zishan.
Moving towards the next presentation, can I please invite Ms. Nikita Yadav? Ms. Nikita Yadav?
Yes. Good afternoon, everyone. I am Nikita Yadav of BSc Honors Chemistry. My participation number is 37. Along with, I have done this project along with my batchmate, who is also participant number 38, Anshika from BStat. So we are going to present our topic together. Our topic is descriptive statistical analysis of River Yamuna in India. So here is the abstract. As we know that it is impossible to overstate the significance of rivers for the continuation of human society. The health of the river majorly depends upon the quality of water. Here, the variations in water quality of a river may be analyzed with the help of some descriptive statistics. In this data, we have taken 5 water quality parameters for River Yamuna. They are pH, dissolved oxygen, conductivity, biochemical oxygen demand, and nitrate. These are taken from 42 sampling stations of River Yamuna, and this has been used for our studies. The data used here we have taken from the report of CPCB under the National Water Quality Monitoring Program of the year 2020. The data has been compared with the permissible standard for water by the Bureau of Indian Standard, BIS, to get an idea about the water quality of river water, on the basis of which further statistical analysis of this data, it can be aided by providing with valuable inferences about river water quality, and thus helping with more efficient policy recommendation and water quality management.
Moving on to the objectives of our study, the Yamuna has been known to be one of India's most polluted rivers in recent years. River Yamuna degradation is mostly caused by agricultural waste, partially or untreated sewage, and industrial discharge. Therefore, a need for various methods to monitor the quality of water and raise the quality of water is created. In this study, we aim to illustrate one such method of quality assessment by using descriptive statistics on the data of some specific water quality parameters which have been mentioned by Nikita for the river Yamuna for year 2020.
Here is the methodology of our project. First is the data collection. The data set used in this study is secondary data taken from the report of CPCB on water quality data of river under the
National Water Quality Monitoring Program of year 2020. The data gave measures on various water quality parameters of River Yamuna through 42 sites covering the whole of India.
The statistical analysis that has been performed and showed in this presentation is descriptive analysis, which is utilized on this data to get an idea about the spatial trend for the water quality of River Yamuna. The data on each of the 5 chosen parameters is graphically represented to analyze the pattern of their change through the various stations. The given data is then compared with the standard values of parameters which have been given by BIS. This gives us an insight about the quality of water and its change throughout India. Here are the...
You are please requested to wind up the presentation.
Here is the summary of the trends that we observed in the various parameters. For dissolved oxygen, the standard by BIS was minimum to be 5. And as we can see, for most of the sites, it was well within the permissible limits, except for a few stations for which it was less than 5. For pH, the water of the stations was well within or around the limit, except for 2 peaks, which were greater than 8.5. The conductivity of water has been standardized to be between 750 to 3000 microsiemens per centimeter. And the conductivity in water was found to be on the higher side of the scale, but still permissible, except for one peak which reached up to 5,866 value. The biochemical oxygen demand was said to be standardized as 10 or as value 10, but the water samples were found to have way higher than the standard value by BIS.
Nikita, please conclude the presentation.
Okay. The nitrate levels in the water were low. This is an ongoing study, and the future objectives that we aim to apply in this study are Spearman correlation analysis with the help of Spearman's rank correlation to study the spatial trend. The same descriptive statistical analysis and Spearman's correlation analysis will also be applied to study the temporal trend for the data given on the site by CPCP. This concludes our presentation. Thank you.
Thank you. Thank you.
So from whatever data you have analyzed, what did you conclude? You have just shown all the samples, some are high, some are low, but what is the conclusion? In just 1 minute, can you just conclude?
Ma'am, the aim of our study was to study what pattern the water of River Yamuna follows through various states and sites that it flows throughout India. So this was a spatial trend of the quality of water, and we have analyzed that it was almost similar, except for a few parameters that reached peaks at some places. And for some parameters as of the BOD and the electrical conductivity, how they were high, and the nitrate levels that were too low with this as compared to the standardized values.
Okay, fine. That's all from my side.
Yeah, I think we can go with the next candidate.
Thank you.
Next up, can we please have Ms. Alka Pandey? Ms. Alka Pandey, are you there? Okay, so moving towards the next presentation, can we have Ms. Palak Mishra?
Hello, am I audible?
Yes, Palak, you're audible.
Palak, start your presentation. Okay.
Good afternoon, esteemed judges and everyone present here. I, Palak Mishra from Amity Institute of Biotechnology, would like to present a presentation on the topic AI enhanced solar energy forecasting, a sustainable path to a brighter future. So, artificial intelligence can be used to optimize and improve the efficiency of solar energy. AI can analyze data, enable process automation, and improve forecasting to make solar energy more efficient and cost effective. AI can also be applied to maximize solar energy output while minimizing operational expense and risk. Researchers are using AI applications to better integrate their solar resources and make more informed planning decisions. Next, artificial intelligence has also been used to predict solar power output. A recent study highlights the use of AI-based models to accurately predict solar power output by considering weather conditions, geographical location, and time of the day. The study used multi-layer perceptron, MLP, and adaptive network-based fuzzy interference system models to forecast photovoltaic power production. The developed forecasting models was trained using historical data from October 2011 to February 2022. The outputs of the processed model were checked for accuracy and compared by considering the data set from a PV power producing station. The MLP and ANFIS models achieved the highest performance with less prediction errors. Next, according to the famous journal Nature, rapid development of renewable energy sources, particularly solar photovoltaic, is critical to mitigate climate change. As a result, India has set ambitious goals to install 500 gigawatt of solar energy capacity by 2030. Given the large footprint projected to meet renewable sources, the potential for land use conflicts over environmental value is very high. To expanded development of solar energy, land use planners will be used to till up to date and accurate geospatial information of PV infrastructure. In this, they developed a spatially explained machine learning model to map utility solar scale projects across India using freely available satellite imagery with a major accuracy of 92%. Their model predictions were validated by human experts, obtained a data set by 1363 solar PV farms. This is the end of my presentation. Thank you.
Okay, Palak.
Yes, ma'am.
I somehow was not able to sum up by the time you said, you have summed
Yes,
up.
ma'am.
But I was wondering, what exactly are you trying to tell through this presentation?
I'm telling that solar energy can also be utilized using AI. Like some of the companies like Sun Power are using AI to generate solar energy also.
But in such presentations, probably we can have anything which you have, initiated or you have taken up. We were looking for that. At least I was looking for that, which I really found was missing. That's all from my side. Aditi ma'am, over to you, and Manish sir, please.
That's okay. That's okay, because it's a hypothesis. It's just
Right.
Okay, Palak, thank you so much. We can move to the next.
Thank you, Palak. Okay.
Thank you, ma'am.
Yeah. So I think we can have next candidate.
Next up, I would like to invite Mr. Divyansh Verma for the last presentation of the day.
Good afternoon, everyone. Am I audible?
Yes, Divyansh, you're audible.
Okay. Is my screen visible?
Yes, it is. Please continue. Just put the screen, full screen mode. Put your slides on full screen. Right.
Good afternoon, everyone. My name is Divyansh Verma, and I am here to present on the topic optimizing biofuel production through artificial intelligence, a sustainable. So basically, what is biofuels? Biofuels are liquid or gaseous fuels derived from the wait a minute. Wait a second.
Yeah.
Wait a second, ma'am.
Don't read each and every word. Just go to the bullet points.
Basically, biofuels are liquid or gaseous fuels derived from biomass, which can be renewable and potentially more sustainable than fossil fuels. The sustainability is the most important part of the biofuels, as it is a critical concern, as they can have both positive and negative environmental, social, and as well as economical impacts. Some key points to consider are reduce greenhouse gases. The biofuels have the potential to reduce greenhouse gas emissions to compare to fossil fuels. And the another is energy balance. The energy input required for biofuel production should not significantly exceed the energy output. If the energy balance is poor, it can undermine the environmental benefits of biofuels. There's also biodiversity and ecosystem health. Sustainable biofuel production should protect and promote biodiversity, maintain soil health, and avoid excessive use of chemical inputs that can harm ecosystems. It also has social impacts. Biofuel, including land right issues, displacement of local communities, and effects on food prices. Sustainable biofuel production should adhere to social and labor standards and ensure local communities benefit from its development. There are many advantages of using AI for biofuel production. For example, enhancing source efficiency and reducing waste, AI-driven analytics optimize resource allocation, minimizing waste and maximizing the utilization of raw materials. That means it helps in the optimization of feedstock. It can analyze data from various sources such as satellite imagery and weather forecasts to optimize the planting, harvesting, and processing of biomass feedstocks. That is why it's also cost effective. It also helps in improving biofuel yield and quality. AI-powered sensors and control systems can regulate various parameters such as temperature, pH, and nutrient levels in bioreactors and fermentation tanks. It also helps in minimizing environmental impact and greenhouse gas emissions by optimizing production processes. AI techniques can minimize environmental footprint and contribute to the reduction of greenhouse gas emissions. It also has challenges and limitations. Data availability and quality issues. Algorithm heavily rely on data quality and availability, which can be challenging due to the complexity and variability of biofuel production. Complexity of biofuel production processes. Biofuel production involves intricate chemical and biological processes, making it challenging to model and optimize using AI techniques. It also has limitations in affordability and accessibility of AI tools. The integration of AI tools and technologies may represent financial and accessibility barriers for smaller biofuel production facilities. In conclusion, I would like to say artificial intelligence has a transformative impact on biofuel production by enhancing efficiency, reducing costs, and enabling development of more sustainable, economical, viable biofuels. These advancements are crucial for addressing the global need to reduce greenhouse gas emissions and dependence on fossil fuels. Thank you for your attention.
Okay. Thank you, Divyansh. Aditi ma'am, any question from your side?
I think that's fine. You have, put a focus on a good topic, which is where AI can be used in biofuel production. So as we are running out of time, I would not like to take more time.
Oh, okay, ma'am. Okay.
Maybe, ma'am, you can ask one or two questions. That is no issue.
No, that's fine. That's fine. Yeah.
Manish, sir.
Okay, Divyas, I think it's good. So we may proceed for the another participants.
No, this was the last one.
Yeah, it is
Oh,
the last.
it was the last one? Okay, okay.
Yeah, you can evaluate it.
Over to you, Prachna.
Thank you to all the participants and the audience. The presentations were wonderful with excellent research contributions. It will indeed be a challenging task for the judges to decide upon the best presentation. The results will be announced shortly. I would now request Director, Amity Institute of Biotechnology, Dr. Janmajay Kumar Srivastava sir, to welcome our chief guest of the day, Professor Dr. Farooq Jamal sir, Head of the Department of Biochemistry, Dr. Ram Manohar Lohiya, Awadh University.
सर किसी दिन में आ जाते
सर पिछले 5 साल के आ जाएंगे.
हाँ, शिल्पी मैम.
Unmute your mic.
Yes. My mic is unmute.
Ritika Tripathi, you are requested to please switch off your mic. Ritika.
Dr. Shilpi. Prachi ma'am. Anybody can listen?
Yes, sir. I can see you, I can listen to you, sir. Both of us.
हाँ बताइएगा, ये आपका वैलिडिटी शुरू होना है या अभी प्रोग्राम आपका चल रहा है?
नहीं sir, we are end with the session. We are end with the oral presentations, and judges have evaluated. And so now we are commencing for the validatory session. Our guest, Dr. Farooq Jamal sir, has joined. So I would like to request you.
Okay. So again, I extend a very warm welcome to Dr. Farooq Jamal, my old friend and my friend from the sister lab as well as the hostel mate. I think we have shared a common time in both the places. So, Dr. Farooq, welcome to this session of for the validity session, and I hope that you will advise some of your valuable points so that in coming time the organizers can improve further in the organizing the events and for the conducting the events. If you have any observation for the organizers as well as our volunteers, as well as the quality of the work which has been presented, and if you have any special suggestion, so I welcome you on this occasion. Dr. Farooq, please.
Sir, please unmute yourself. Sir, please unmute yourself.
Am I audible?
Yeah. Now you are audible.
Now you are.
Thank you, Dr. Srivastava, for introducing me to this gathering. And yes, we have been a long-time friend. We have been working in a lab which was very associated with your lab. And then we were in the hostel for some time. And I have visited your university quite a number of times. So this validatory session has already laid. I must say that there are two prominent things that should be kept in mind. One is we should be very, we should control the time, and it should not be extended to because everybody has got some sort of planning. And being in an educational institution, you never know when a meeting is to be held. And anytime we can be called by the Vice Chancellor for some meeting, because two great occasions are in the offing. Number one, we are going to have this Deeputsav, in which huge number of diyas has to be lighted on the occasion of Deepavali. So around 21 to 24 lakhs of diyas are going to be lit on the 11th of November, which is again going to create a world record. And the next thing that is going to come after Deeputsav is the convocation that is scheduled for 29th of November. So two big programs are in the offing, and that is why anytime the Vice Chancellors call us for meeting. So now I welcome this August gathering in the validity session of this 2 days international conference on high throughput data analysis and artificial intelligence in biological research, which is being held online from the Lucknow campus of Amity University. I am privileged to chair this closing session, and at the outset, I am exhilarated to congratulate the convener of this successful conference, Dr. Prachi Srivastava and Dr. Shilpi Srivastava, who were instrumental in executing the script of this conference quite smoothly and effectively. I am grateful to Dr. Prachi Srivastava for inviting me to chair this session. The organizing secretaries, in association with the panel of advisory committee, did a commendable job to bring in delegates who were distinguished speakers on the present theme. I acknowledge the chairperson, Professor J.K. Srivastava, for his dedication in holding the audience and keeping the enthusiasm alive with the resource persons without giving any feeling of dryness on the theme of today's expression. Also, I am happy and grateful to the founder president, Honorable Dr. Ashok K. Chauhan, Chancellor, Dr. Asim Chauhan, Pro Vice Chancellor, Professor Anil Kumar Tiwari, Dean Research, Science and Technology, Professor Kamar Rahman, and Dean Academics, Professor Rajesh K. Tiwari, without whom the international conference would not have materialized successfully and with extraordinary enthusiasm. Also, I am glad to know that Professor Anu Chandra from the ERA University too has been a part of this conference as the chief guest of the inaugural session. Friends, the conference was exemplary. It went very well. But I have certain opinion regarding the science, the technology, which is my own personal opinion. And these things have come in these around 20 to 25 years of teaching and research and being in the field of science and technology. Undoubtedly, in recent decades, science has made surmountable and significant progress in all dimensions. Biological science has made some exemplary progress in translation research. Data mining tools have been an asset in processing the data and finding out if the work is relevant, substantially significant, and beneficial for mankind. High throughput, as the title of the conference is, data analysis is rapid and sufficiently accurate approach of handling large samples using automated processes. We are witnessing an era where technological revolution is taking biological sciences to a flashpoint after which translation of revolutionary ideas seems to be streamlined. Robotics and artificial intelligence are replacing humans in taking up challenging tasks and resolving them at breathtaking speed. Now with a gadget, so-called mobile, every human can access any repository of information and can connect, coordinate, and orchestrate virtually any activity, thus forcing heavy dependence on it. That day is not far when an evolved and organized mass of cells called humans will be servant of technology and world may be inhabited by species which lack all characters of so-called beings. Despite being somewhat prejudiced on technological front, as we are all Homo sapiens, some wonderful deliberations have rocked the technical sessions. Many novel inspirations, many novel impressions also have touched the young scientists and research scholars who may aspire to move ahead with some breakthrough research. Undoubtedly, modern problems, be it local or global, requires modern solutions. Obviously, the technological breakthroughs in coordination with artificial intelligence will pave a way for a better and not bitter future for the existence of life on this planet. Due to time constraint, I would request the technical team to flash my PowerPoint presentation so that I may quickly summarize the theme of the conference for those who felt that contemporary issues require some simplified version of explanation. So I can see my slide, and let me tell you that I was witnessing most of the oral presentations. The presentations, as far as the slide was concerned, it was excellent. As far as the oration skill was concerned, most of the students, research scholars, they presented very well. And for the first time, I've seen that the participation from the angle of undergraduate as well as postgraduate students have been remarkable. This is something that needs to be instilled, inculcated in other educational institutions. And a big round of applause for the MIT University that it takes such issues and tries to help the students, motivate the student, encourage the students for presentation, for giving them an opportunity to present what they think. The idea may be rubbish, idea may be novel, idea may, it can oscillate between two extremes, but at least they are coming up with something which could be which could help the science or which could help them to progress a lot. As far as the judges were concerned, I find that most of them had did the hard work and they were consistent as far as asking the questions were concerned for each of the students who presented their work. And I must congratulate them for their patience. And I think this is something that that inspires the students, that encourages the students. I acknowledge them, and a big round of applause for them too. Now, since this was a 2-day international conference, and many renowned speakers joined and presented their specific areas of research. And this is not the right time for me to take up the issue on high throughput data analysis and artificial intelligence, as you know, many lectures and many things have been talked about how this throughput data analysis and artificial intelligence can go a long way in paving new ways to biological research. So I will just try to give an overview about what is this high throughput and data analysis and what is this artificial intelligence. Perhaps when the generation down the line has come to know about the impact of artificial intelligence, they are talking about its application in various areas of science, be it solar, be it pollution, be it medical, be it everything, anywhere you can apply this this artificial intelligence and this high in any areas of life. So I'll just rush through a few of the slides. I would request to move on to the next slide, please. Oh, okay. This high throughput screening, as the name suggests, is a method, is a way to identify one or more positive candidates, which are extracted from a pool of possible candidates. Now, what some of the speakers have said, that it is very data mining and data and drawing out important datas, important conclusions from the datas is a big task. How to handle such such huge data? So here also you see that high throughput screening. You know, the aim is to find out the hit or the leads that affect the target in a desired manner, active at fairly low concentration and more likely to show specific. So some of the techniques which are which are used. Next slide, please. These are the detection methods in HTS, and most of us are familiar with the techniques which are which are being used in HTS: spectroscopy, mass spectroscopy, microscopy, radioactive methods, and so many other. Next slide, please. So even in the spectroscopy, you will find that there are many different types of spectroscopy: fluorescence, light scattering, Fourier transform, infrared. Then there are chromatography in HTS, and I think most of us are familiar because we are all from the biological sciences. So this physical techniques like gas chromatography, thin layer chromatography, liquid chromatography, all these are, you are all familiar with them. Just I'm presenting an overview of it. Next slide, please. Next slide. Then let's come on to the methodology part of it. And we all know that since a huge amount of sample has to be screened in no time, so accordingly accordingly you have to configure the plates. And it has moved from 96 wells to 364 and now to a high density 1536 well format, which enables large scale screening. This is the basic advantage of it. Like a huge samples can be screened in no time. Next slide, please. Now, this is just to give you an overview, like the reagent is coated on the well or added in liquid form to react with the samples, and then samples and reagents interactions are detected. All these things are, have become very too common. We have moved much ahead of these. Next slide. Next slide, please. Okay, now cell-based assays have become important tests compared with other in vitro assays, as they can provide information about bioavailability, cytotoxicity, and effects on the biochemical pathway. The enzyme-based and the cell-based, assay system consists of receptors or mimetics or receptors, which are components that mimics active parts of the receptors. Next slide. Okay, move on to the next slide further. So this is just a representation which I have picked up from somewhere. And you can see that there is a conventional method of assessing the sample, and there is a high throughput method of assessing the sample. In high throughput, you see the huge number of samples can be screened, and you can get the results in no time. So high throughput screening these days are solutions to modern problems, what you can say. Next slide. This drug design, we all know that in pharmaceutical research, high throughput screening is highly required, because number of candidate molecules are there, and the best molecule which is going to be effective is to be screened out, and for this some scheme is designed. Next slide. I have talked about the cell-based assays, and in the cell-based assay, you see variety of assays that measures the cell proliferation, toxicity, motility, production of measurable amount. What happened? Okay, can you enlarge the see? Okay, no problem. There's a cell-based assays offer a more accurate representation of the real life model. You see the cell, if you are working on the cell directly, that means the results can be more fruitful, because ultimately the aim of any objective is to find out some molecule that would really do some wonders on the human system. So cell is very close to the it is a part of the human system, and if you're working on the cell, it will be much more beneficial as compared to in vitro studies. Next slide. There are 4 key elements of the cell-based assay, and you need a cell line or a primary cell population, and then there are target molecules that records the cellular response, an instrument to conduct and monitor the assay, then informatic components to manage and analyze the data from the assay. Next. I think with this you are all familiar that cell-based reporter assays are used when human receptors are transfected into null cell lines, either alone or as a part of receptor systems. And next slide, please. Okay, you move on to the next slide. So some examples of the cell lines are, you see human cell lines, then there are different cell lines which are being used. Acute myeloid leukemia, these are cell lines. Work on cell lines have gained popularity these days because it mimics the natural system. Next slide. Now, applications of high throughput data analysis, you can see virtually it is working in all the fields, in all the areas, right from genomics to whole exome sequencing, transcriptomics, ribosome profiling, then proteomics, protein and RNA structures, systromic, then genome architecture. You see, everywhere it can be applied. Like some of the presentations talked about using this artificial intelligence anywhere. So both these things, high throughput data analysis and artificial intelligence, both are somehow interrelated, and both are going to change the entire face of the science. Next slide, please. And this I've just given you to see that in genomics, mutations, gene duplications, genome rearrangement, gene regulation networks, and phylogenomic relationships. And in epigenetics, you can see the histone modification, DNA methylation. DNA methylation, it's an important phenomenon. It can, silence the gene, it can activate the gene. And in transcriptomics, differential regulation of the gene. It is very important to understand how a gene is regulated, how the gene functions. So, next slide. So even let's okay, that's fine, no problem. So we move on to the artificial intelligence, about which there have been so many talks, and this will cover up, I think, the entire story of this 2-day conference, that this artificial intelligence has become so prominent that it has entered into various aspects of agriculture, various applications in biotechnology, like industrial biotechnology, medical biotechnology, animal biotechnology, and it is simply surprising that with the artificial intelligence, this I have told you earlier, in years to come, you will see that humans will be replaced by robots. And this is happening. I came across a news in the Hindustan paper. Even it was there in the Hindu paper, that a school was being run by a robot. Means the principal was replaced by a robot. So how is it possible? Is it possible only through the artificial intelligence? And moreover, you see that post-corona, you see, most of the things are being held online. There is no need to visit. We are all connected. This entire population, whether somebody is sitting in the United States, somebody is in Sri Lanka, or somebody is in Japan, all of them are connected. So कहते थे ना कि दुनिया जो है वो सिर्फ एक पूरी मुट्ठी में है ना, वो मुट्ठी में आई गई है. And similarly, this in times to come, you will see that artificial intelligence, which has percolated, all the spheres of human existence, it will transform or transmogrify the human beings into, some machines. Next slide. I see. This is just to highlight for those who are unaware, like in animal biotechnology, prediction of the growth rate, some aspects, optimization of feed efficiency and energy intake, prediction and prevention of diseases. In medical biotechnology, we are well aware that high throughput has been a very good source, a very good tool for drug discovery. And in healthcare systems analytics, artificial intelligence is going to change the entire scene. Then precision medicine is there. And industrial biotechnology, you have intelligent biomanufacturing, designing of bioreactor for optimal yield. And agriculture, you see precision farming, monitoring of soil and crop health. Next slide, please. Next slide. Well, this was simply just to give you an overview and connect you with the impact of high throughput data analysis and artificial intelligence that is being much talked about. And you can see the same thing has percolated even in the human beings, that yes, we are turning into robots. We are becoming more mechanical rather than retaining the feelings of the human being. Well, let's see what happens in days to come. So I think I'm already late. I have to attend a meeting. And once again, I congratulate the organizing committees, the technical committees, and all the presenters who did a wonderful job in coming out and presenting their ideas. Some of them presented the ideas itself. I also like that, because at least they're coming up with some new ideas on which no questions can be asked. That is also, that is also a way of expressing your scientific temperament, your scientific, knack. So, and it has been a wonderful 2 days interaction, and hope that such conferences will be organized in near future. And MIT University is doing well on this front. And once again, my congratulations to one and all, and my blessings to all the young budding scientists who have come here, and all the students as well who have presented the things. Thank you very much. Thank you.
Thank you so much, sir, for your significant talk on high throughput data analysis and artificial intelligence, paving new ways for biological research. Surely, high throughput data analysis and artificial intelligence are new age approaches that refine research and development. I would now extend my heartfelt invitation to Dr. Aditi Singh ma'am to end the anticipation of the participants and announce the results of the presentation session of both the days.
Thank you so much. Thank you so much, the Master of Ceremonies. And before declaring the results, I must congratulate all the participants for both the days. And there have been very good deliberations on day 1 as well as day 2. But I'll not take too much time, and I'll simply go towards the results. So for day 1, or the technical session 1, I'll start from the 3rd position. So the 3rd position went to R. Bharti Ratnam, and the institution has not been given in here, but R. Bharti Ratnam got the 3rd position. For second position, there's a tie, and Anugya Srivastav from CSIR IITR and Anurag Singh, CSIR IGIB, got the second position for first day technical sessions. And the first position for day one went to Ms. Shweta Singh Chauhan from CSIR IITR. So, big congratulations to all the winners. And moving to day two, technical session 2, the third position has a tie, and there are 2 winners for position 3, for which we have Palak Sachdeva from Amity University and Muskan Gupta from BITS or BITS, etc. Congratulations. And at second position, we have Zishan Arif from CSIR IITR and Mihir Tandon from Amity University. And for first position, we have Mohammad Talha from Amity University. And to all the achievers, my lots of congratulations and best wishes for future. Over to you, Ananya.
Thank you, Aditi ma'am. Thank you so much.
Congratulations to all the winners. Your outstanding research and unwavering commitment to the field have truly paid off. We also recognize the tremendous effort and talent displayed by all the participants. Thank you all for being a part of this conference and making it a huge success.
I would now request Dr. Shilpi Srivastav ma'am to give the concluding remarks of the day.
Thank you, Sanjana and Ananya. So good afternoon, one and all present here. With the blessings of Almighty and seniors, we are here at the end of 2 days international conference. It is my profound privilege to deliver the concluding remarks of the International Conference on High Throughput Data Analysis and Artificial Intelligence in Biological Research, held on 19th and 20th October 2023. The title of the conference is of high significance in the current scenario. The day one of the conference was started with the inaugural session with the presence of eminent dignitaries, Professor Dr. Anuchandra ma'am, Registrar, ERA University, Lucknow, Dr. Farooq Jamal sir, Head Department of Biochemistry, Dr. Ram Manohar Lohia, Awadh University, Awadh, our own Pro Vice Chancellor, Wing Commander, Professor Dr. Anil Kumar Tiwari sir, Dean Academics, Professor Dr. Rajesh K. Tiwari sir, Director and Head, Amity Institute of Biotechnology, Professor Dr. J.K. Srivastav sir, and blessings of the Dean Research Science and Technology, Professor Dr. Q. Rahman ma'am was received, which was delighted on conference. The inaugural session was followed with invited lectures, and the session was chaired by our dignified researchers, Dr. Jyoti Prakash ma'am and Dr. Ruchi Yadav ma'am, where we had some eminent speakers from across the globe. The session started with the invited lecture of Dr. Anshul Tiwari sir, scientist from Molecular Physiology and Biophysics, Vanderbilt University, Nashville, USA, who delivered a very interesting and captivating talk on the role of artificial intelligence in electronic health record. The session proceeded with another educational and innovational talk of Dr. Khurshid Ahmed sir, Research Professor, Department of Medical Biotechnology, Yongnam University, Republic of Korea, and who delivered his talk on one of the most trending topics, that is, exploring natural compound as potential 5-alpha reductase type 2 inhibitors, a biocomputational investigation. After such interesting talks from the experts, we had the second technical session where the young minds presented their research findings and thoughts. And it was evaluated by our knowledgeable judges, Dr. Mala Trivedi ma'am, Dr. Saili Mukherjee ma'am, Dr. Vineet Awasthi sir. The day 2 of the international conference was started with the technical session 1 through invited lectures. Session was chaired by our commanding researchers, Dr. Rachna Chaturvedi ma'am, Dr. Meenakshi Srivastav ma'am, and session was started with the introduction to a very new and latest topic on ribosome sequencing by Dr. Srinivas Subramaniam sir, Associate Professor, Department of Neuroscience, the Scripps Research Institute, Florida, who delivered his talk on ribosome profiling and mass spectrometry reveals widespread mitochondrial translation defects in skeletal cell model of Huntington disease. Moving on, we had an invited lecture from the senior scientist, Dr. Jiten Narayan sir of ICHTDAAIBR New Delhi, who presented a very inspiring talk on AI-powered diagnostic system for neurofibromatosis. His talk was not only informative, but currently one of the most demanding field also. Next, we had very interesting talk by Dr. Sapnil Srivastav of Medbug Cardiac Clinic and Heart Hospital, Mumbai, India. The talk was focused on AI-based approaches to improvise healthcare. After the mindful thought of the eminent speakers, we move forward towards the second technical session. That was oral presentation, which was evaluated by our eminent judges, Dr. Gurjeet Kaur ma'am, Dr. Manish Bedi sir, and Dr. Aditi Singh ma'am, where we explored some of the recent studies done by the students around the world. Last but not the least, we had one of the best valedictory sessions from Professor Dr. Farooq Jamal sir, Head of Biochemistry, Dr. Ram Manohar Lohia, Awadh University, Ayodhya, who focused on the application of artificial intelligence and high throughput data analysis. He summarized the main essence of the conference very well through his talk. These 2 days conference were indeed a knowledgeable, informative, and exploring session for all the participants. I hope you all found something to take back from the conference and implement in your research finding also. I am happy to announce that we had 65 presentations from different institutions of India with CSIR, ICAR, IARI, BITS, SIFI, Bombay, Narayana Institute, Era University, Amity University, SRMU, Chandigarh University, AKTU, and Jamia Millia. Presentations were excellent and shows the success of the conference. So thank you all. Now I would like to invite Dr. Prachi Srivastav ma'am for the vote of thanks. So ma'am, please.
Thank you, Dr. Shilpi. So I know all of us are getting late, but it is my profound privilege and honor to propose a formal vote of thanks among you all. So first of all, I extend my very heartfelt thanks to Almighty for his blessings. Because of that, this virtual conference has taken a proper shape. Proceeding ahead, I am really thankful to our honorable guests, Professor Dr. Anu Chandra and Professor Dr. Farooq Jamal, for their gracious presence. I extend my very heartfelt thanks to our eminent speakers for all the sessions. Dr. Anshul Tiwari, Dr. Khurshid Ahmed, Dr. Jitendra Narayan, Dr. Srinivas Subramaniam, and Dr. Swapnil Srivastava for their excellent and insightful deliberations given on this virtual platform. Really, it was so interactive that is evidential from the question answer sessions. Thank you. I am thankful to Dr. Ilyas also, though he couldn't join the podium because of his emergency of health issue. Yet he had sent his talk summary and messages to us. I am dumb to express really my gratitude for our founder president, Honorable Professor Dr. Ashok Chauhan sir, our Chancellor, Professor Dr. Atul Chauhan sir, our Chairman, Professor Dr. Asim Chauhan sir, our Vice Chancellor, Professor Dr. Balwinder Shukla Madam, for their vision towards such advanced applied areas. We are always motivated by their blessing, with their visionary thoughts and their motivation. I am heartful thanks. I will be delighted to present my thankful to our own Pro-Vice Chancellor, Sir, Wing Commander, Professor Anil Kejriwal, our Dean Academics, Professor Dr. Rajesh Kejriwal, sir, for their consistent mentorship throughout the journey. I am blessed to receive the message of our Dean, Research Science and Technology, Professor Dr. Kamar Rahman Madam. Thank you so much. The success of conference cannot be achieved without the consistent guidance, support, and visionary thoughts of our own director, Professor Dr. Janmejay Kumar Srivastava. His consistent support, guidance, with unconditional reasons throughout the start and end of the conference was a pillar of the success. I am extremely thankful to Professor Dr. Colonel Vivek Mathur sir for providing all kind of necessary support when and where required. I am feeling nostalgic in expressing my thanks to our chairs, Dr. Jyoti Prakash Madam, Dr. Ruchi Yadav Ma'am, Dr. Rachana Chaturvedi Ma'am, and Dr. Meenakshi Srivastava for chairing the technical sessions throughout the conference. No doubt, Meenakshi was my technical advisor too. Thank you to all of you. I do not have sufficient words for expressing my deepest gratitude towards my eminent judges, Dr. Vineet Avasthi sir, Dr. Sally Mukherjee, Dr. Mala Trivedi, Dr. Aditi Singh ma'am, Dr. Gurjeet ma'am, and Dr. Manish Trivedi for their continuous listening of oral presentation and motivating the students throughout their visionary thoughts and their experiences. They asked such a relevant questions from the speakers and they judged the session so nicely that I do not have words to express my gratitude. Thank you to all of you. I owe my thanks to the advisory members and all faculty members of KIB and staff and other institutions for their support and encouragement in the form of their e-presence. I would like to further extend my gratitude toward all participants who joined us for this last few days. As without participants, there is no achievement, there is no conference. It is indeed their active involvement that has made this conference a great success. Thank you to all of you for being with us. Any event cannot take a proper shape without collaborative efforts. So I extend my very heartfelt thanks to my co-partner, Dr. Shilpi Srivastava, for a consistent contribution towards the event. I cannot stand without my volunteers. That's really from my core of heart. Aman, Ananya, Vais, Sanjana, Prekshi, they are my backbone on which I can stand on and I can perform the things in such a planned and executive way. Their tireless and endless efforts throughout the journey is truly commendable. I am really dumb to express my gratitude to all of them. So, the conference would not have been possible without the technical and unconditional support of Director IT, Mr. Gaurav Mathur sir, for providing us with the Zoom platform at such a short notice. So thank you so much, sir. I am also thankful to Mr. Momin from Noida campus. He was throughout the conference and pursuing the technical problems and taking the session in such a proper manner. I'm thankful to my Surendra Bhaiya, Sunil Bhaiya, Jitendra Bhaiya for providing us all related support to us in bringing tea and coffee whenever required during the execution of this e-conference. Last but not least, I would like to take my apologies if anybody is left during my thanks remarks, but still from the core of my heart, I extend a very warm thanks to all of the presentees in this conference. And once again, thank you so much for all the support you have shown to make it this conference a good success. And I am really taking the words of our guest, Dr. Farooq Jamal, the session should be in the time limit. So though we tried to be in the limit and it is the 17:25, we wind up the session and tried that we have not bothered you more. Thank you so much, sir, to all of you. Thank you. So now if it will be nice if we can have 2 minutes for our national anthem. Thank you so much.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
Thank you to all of you. Now we can leave the session. Thank you.
I express my gratitude to each one of you for being so cooperative and making this conference a huge success. The e-certificates and the abstract book of the conference will be shared to you with mail shortly. Thank you.
Thank you, ma'am. It will be nice if we
Congratulations
take.
to all for a successful conference and event. All the organizers, ma'am, Meenakshi ma'am, Prachi ma'am, Shilpi ma'am.
Thank you so much, Aditi ma'am, for being with us. And
Very
blessing us.
nicely, very well organized conference, Prachi ma'am, Shilpi ma'am. Aditi ma'am, your questions were excellent. Huh? I was there, I was listening. Excellent questions by the judges. Yeah, yes.
Are truly
No
admirable.
doubt.
Truly
Matlab,
admirable.
one after one, they keep on listening everything so
Everything.
seriously, and then the
And
questions.
such a relevant questions they were asking.
Yes,
That's
true,
very
true.
nice.
Can
Really
we have a
mind
group
from my bottom.
photograph, Prachi ma'am, for the report?
How can we take by opening the cameras?
Yes.
Okay.
All those who are present can open their cameras.
Yeah. Mostly the winners. Winners, let's have a sweet memory with you. Please switch on your camera.
Open your camera.
So Anugya, Jishan,
Your student has won the first prize today.
who's
Meenakshi
ma'am? Meenakshi ma'am.
Yes.
I was just asking, is your student got the first prize today?
I don't know, ma'am. What was the name?
Name क्या है ये लोग बताएंगे. Ananya will tell what is the name of today's winner.
Mohammed Talha.
Talha.
Yes,
हाँ.
he's the student of MCA. Yes. And who was the second one? Ananya?
Ma'am, Mr. Mihir Tandon and Sir Shan Arif.
Okay, good.
So Zishan Arif
Congratulations,
from IITR,
students.
and the other one is also from Amity, so maybe from your institution only.
Yes.
I really like the work of
Congratulations,
Talha.
winners.
They are not
Students, switch
here
on the
right
camera so that we can have a quick group photographs.
Just
Everybody
a minute.
is.
I am sending the message on the participants
Yeah,
group.
Mihir is there. Mihir, congratulations.
Beta participant group.
Thank you, ma'am. Thank you so much, ma'am.
And just implement all the suggestions which has been given by the ma'am.
For sure, ma'am.
I know we are getting the.
Yes. Okay, Ananya, are you taking the pic or should I take it?
Ma'am,
The
I'll take
screenshot.
Bas
Okay.
Meenakshi ma'am, 1 second, one more participant is opening.
Okay, okay,
Anugya.
sure.
Congratulations, Anurag, Mihir.
Yes, congratulations.
Thank you so much, ma'am.
Well
Thank you so
done.
much, ma'am. Thank you so much. Thank you, ma'am.
Anurag is there and it is bound to win. Huh? He is bound to win.
No, ma'am. Ma'am, all due to your all efforts. Whatever I have learned, it started right from there only, from Amity Institute of Biotechnology. So I owe you all a big thank you.
Anugya, congratulations. Anugya is there. She is a
Yes,
winner.
Anugya, congratulations, bache. Well
Thank you,
done.
ma'am. Thank you so much.
Okay.
Mihir
Now,
and
can we
one,
take a photograph and then we can
Yeah.
leave?
Please.
I think Jishan is typing his just opening. So Jishan, but then we can take, okay. Otherwise, Jishan, are you opening your camera?
Wise, please switch on the camera. Ayushi, Wise.
Why is it sitting together? No?
Okay, Wise is there. Okay. So Ananya, please take a screenshot.
Ayushi is there. Ruby ma'am can
Dr.
open the
Khurshid,
camera.
I am thankful to you. You are on the dais. If you are listening me, will you please open your camera in such late hours?
Thank you so much, ma'am, and many congratulations to all of you.
Yeah. Thank you so much.
Well done.
Ma'am, I've captured the screen.
Thank you so much.
Thank you. Well done, Bachu. Well done, team.
Thank you, ma'am.
Thank you so
Thank you
much,
so much.
ma'am. And congratulations to all the winners.
Thank you.
Thank you. Thank you.
Okay, Prachi ma'am, do we
Thank you
have
so much,
your
ma'am. And
do we have your
congratulations.
permission to leave the team meet?
Bilkul. I tried.
Thank
I
you.
tried to club up in the time. Thank you so
Thank
much
you.
to all
Take
of
care,
you.
everyone.
Thank you, ma'am.
Bye-bye.
Thank you, ma'am.
Bye.
Bye.
Thank you.
Bye, ma'am. Thank you so much.
Thank you, Bachu.